morning, everyone. Good morning, people here at the beautiful Aula Hall at the University of Iceland, and you people everywhere, wherever in the world you might be watching this seminar. I'm Tuija. I'm the chair of the Nordic Committee of Bioethics, and on behalf of the committee and its members, I would like to warmly welcome you all to the symposium on the moral status of stem cell-derived embryo models. This symposium was overall made possible by the support of Nordforsk and the graduate program of the BioMedical Center, the University of Iceland. And many people have been instrumental in making this symposium happen. It's always a big deal to make events like this happen, especially now that we are doing streaming and everything like this. But because we thought that this is such an important matter, we wanted to make it accessible as many people as possible. Of those people that have been really key to make this event possible, I would like to thank Bettina, whom you can't see, who's up there, who's on behalf of Nordforsk being responsible for basically all the background organization of making this happen. And then there's another person, Guðrún, the chair of the working group for this seminar, and on whose suggestion we actually originally, the Nordic Committee of Bioethics, decided to go with this particular topic. Since I'm a philosopher and not a scientist, and we need the science background to overall understand these things, I think without further ado, I will ask Guðrún to come up here to explain you a little bit about the topic, about what's going to happen today, and also if there's any practical matters in terms of that you need to be aware of during the day. On my behalf, just enjoy the talks. I'm sure they'll be very thought-provoking, illuminating, and good. Thank you.
Thank you very much, Tuija, and good morning to you. My name is Guðrún Valdimarsdóttir, and I will be the chair of this mini symposium today. As Tuija said, this is a joint collaborative effort between the NCBio and the graduate program in molecular life sciences at the BioMedical Center. And the aim of this symposium is to get insight into the different perspectives of stem cell-derived human embryo models. So by focusing on the outstanding challenges in this new field, and this we do both with technical aspects and ethical aspects. And we get experts in these two disciplines to do that. So the practical things that I want to mention, I will raise a red flag when there are 2 minutes left of the speaker's talk, and I will stand up if the time is really out. So, and after each talk, we will have some time for a couple of questions, and we will have people in the audience who will then bring a microphone to you if you would like to raise some questions. So, with that, I am honored to introduce the first speaker today, who is Dr. Jakob Hanna. He is a principal investigator at the Weizmann Institute of Science in Israel. He is one of the pioneer scientists in the field of embryo models. And indeed, last week, he published a pioneering article on the 14-day-old human embryo models. And they are derived from naive embryonic stem cells. And he will be hopefully telling us more about that. So with this introduction, I would like to give a warm welcome to you, Dr. Hanna, and ask you to come to the podium, please.
So, good morning. Thank you very much for the kind introduction. I'm really happy to be here. my first time in Iceland, and also really look forward for this today, which is continuation of yesterday's discussion, and really hear feedback and hear people's thoughts as well on these kind of topics. So what I'll do is a bit try to talk from stem cells to embryo models and try to explain why we are doing this and how did we get there, because I think it is relevant to really understanding, influencing a lot of the talks about ethics and regulations of this field. So as you know, we study pluripotent stem cells. Typically, originally we started with ES cells, which are embryo-derived. But I'd like to remind us all that pluripotency, unlike adult stem cells that we have the skin or the blood where they just continue to maintain our tissues throughout life, pluripotency is a transient state that exists for 1 day in the mouse, maybe 2 or 3 days in humans, in the inner cell mass. So therefore, I would argue that ES cells are a very useful artifact, and we keep them in the culture by giving them the right cytokines or small molecules. Major one, for example, is the blocking of FGF signaling in mouse and so forth. And then we had a new player on the block, which is iPS cells, which is reawakening of pluripotency in development. And there's a lot of questions, of course, on how we use it. But the main advantage is that we now have genetically identical pluripotent cells on the table. And I would say overall today, the technology, I think we can make iPS cells that are basically indistinguishable from embryo-derived cells. But what we've learned over the years, I work with different groups, that pluripotency is not a binary state of just 0 versus 1, that actually it's a decay process towards differentiation. So you'll have different flavors of pluripotency. So the ES cells originally derived in mouse are embryonic stem cells, correspond to the inner cell mass in the blastocyst stage, which is pre-implantation. And the terminology the best term really put by Austin Smith is to call it naive pluripotency. But actually pluripotent cells after implantation, and here you see the egg cylinder shape typical of the mouse, you also have OX4 positive cells, which is the marker of stem cells, and we can derive these cells, which are much more flat. They're called epiblast stem cells, but now we refer to them as primed stem cells. So they're not differentiated yet, but they are primed to differentiate, and they have different qualities. And this really suggested that at least we can think of this as 2 attractor states, that stem cells can be in at least 2 states. And what dictates this state is the growth conditions, meaning if you give a certain set of cytokines, you keep the cells in the pre-implantation state. If you change to a different set of cytokines, they will just differentiate a little bit to a prime state. And you can switch back, actually, and will revert back. So it's about the media. And so you have cell type, which is ES cells and iPS cells from the embryo, but the cell state is about the media that you give. So you can have naive iPS cells, you can have primed iPS cells. And over the years, again, us and many, many other groups really dwell deeply on these differences. What is the metabolic differences? What are the gene differences? What is the DNA methylation status? And there are a lot of molecular differences between these states. Sometimes they're even like really an opposite. But there was a couple of questions about this. First, what was going on with human pluripotent cells? So the human pluripotent cells originally isolated, as you can see in the lower right part, they were very similar, although not identical, to the mouse primed stem cells, both in their morphology, this flat morphology, epithelium, and depending on FGF signaling, very similar. And for years it was assumed that, well, we should accept them for what they are because we derive them from the blastocyst. They must reflect the blastocyst. It must be the millions of years of evolution differences. That's why they are different. But as I told you, what dictates the state we capture, this artifact, is the cytokines, meaning we could have a scenario where what you start with is not necessarily what you get, that the blastocyst just differentiate towards this lower attractor state because of the media we were giving it. So the question was, are there naive cells, meaning do we need different conditions to capture naive cells in humans? What are naive cells in humans? Is it a rodent-specific phenomenon, or maybe it exists in vivo in humans, but we will not be able to capture it? A lot of questions. But we knew that also the mouse naive conditions do not work for humans, so they're not sufficient. So just briefly, 10 years ago, my group started looking at, can we make naive cells? And for us, the first differentiated between these different flavors of cells is that naive cells, if you block MYC FGF signaling, they will prosper and they'll stay in this naive neighborhood. Versus primed cells, if you block ERK signaling, they will differentiate. And through different calibrations, we found out that we can take the naive conditions in the mouse, this ERK and Wnt stimulation, just add more inhibitors and add activin A, which is surprising, but I'll not get into this. And you can get cells that are different, they are ERK independent, and we termed them at the time as naive. I guess today let's call them more like naive-like, maybe formative, but they're more earlier in development. This media is commercialized, it's a conflict of interest, but at least we started seeing that we can make alternative states in human. And after that, it was still a challenge for the field that we knew that these naive cells do not have features of the inner cell mass, so we don't have everything. And there was a question, what is a better naive conditions? And different group has really excellent media with pluses and minuses, but for us, we wanted to make an enhanced media, which we called Hansen. And just to talk about challenges in embryos, we were facing with a challenge, how do you really prove that a cell is naive? What is the functional characteristic in humans? Because in the end, in the mouse, I would say the proof, if you have a stem cell, you inject it in a blastocyst, and you can make a mouse out of it, that's quite enough proof that it is a naive stem cell. However, in humans, of course, we can never do this, and we should never do this. So there is a challenge. And here, actually, just touch on what is naive pluripotency. When we were working on mouse stem cells, we were asking, are there specific features of the primed pluripotent, which wasn't known at the time? And we did like these knockout of different genes in naive and primed mouse cells, which both have this OCT4 GFP. OCT4 is a marker for stemness. And we wanted to look, are there genes that if we knock them down, the naive cells are fine, but actually the primed cells deteriorate. So the primed cells are in the blue here. And you can see these genes in gray, all of them, if we knock them down, even knock them out, naive cells are completely fine, but the primed cells deteriorate. So there are such genes. And if you look what's common between them, they're all some kind of repressor. You have DGCR8 and DICER, they make microRNA, which is a repressor. You have DNMT1, which makes DNA methylation, which is a repressor. You have METTL3, which is an RNA methylation, it's a repressor, and so forth. So in a way, if I need to a little bit philosophize that is a bit suggesting that the naive state is a very basic state. Although it has these machinery, it doesn't need them. It doesn't need to have these repressive machinery. And if I want to make like, as if it's more akin to a very basal state, to a unicellular organism like yeast. So yeast doesn't have microRNAs. Yeast doesn't have DNA methylation. Yeast doesn't have mRNA methylation. So it's really this basal state that, although in my mind it has them, but to survive it doesn't need these machineries. So anyways, this is just kind of conceptual thinking about if this is really the building state in development. Because typically we think of the building state as the zygote. So I will get to that a bit later. So just to say that for us, this was a test to make human naive stem cells. So we wanted to make now human naive cells that can tolerate loss of DNA methylation and screen for that condition. So we made systems where we take, this is the RNA methylation enzyme. We made a system where, if we give doxycycline, we shut down the expression of this gene. And we screened for conditions whether these cells exist. And indeed, we took our NHSM conditions, we added Wnt inhibitor and Src inhibitor, doesn't matter the details, and now we have cells that if we want, we can remove these enzymes and they will still survive. And really we saw that they much more resemble the inner cell mass in the human embryo. It's actually also resembling a little bit before that, the molar stage, but we'll not get into that. So bottom line, in humans, I would say we have now a variety of different conditions of different degrees of naivety that exist, and we can grow them in xeno-free conditions and everything. And then, personally, I would say I made my tenure and faced this challenge then as like, who cares? Why does it matter, really? Okay, all these differences. You can go all day long back and forth between naive and primed in humans and mouse, but what does it really matter? Or it's of a larger concept. What I'm suggesting, that we now know that the pluripotency has different states. It is evolutionarily conserved. Why is it evolutionary? What is the benefit of it? And I have no answer yet. But there is proof that it does matter. And I'll say a very prominent example is the work from Hayashi in Japan, who was trying to make germ cells, or PGCs, that are going to become sperm and egg later in life, from ES and iPS cells. And he was the first really to make authentic ones in mouse, and his secret for success that he had to start with these naive conditions that have blocking for ERK inhibitor. He had to give them priming with FGF activin, but only for 48 days, which is what happens in vivo, but not longer than that. If he gives longer than that, the efficiency drops to 0. And then he pulses with cytokines, mainly BMP4, and you will get these double positive cells, which he proved that these are germ cells. If you inject them into an ovary, they will make oocytes and so forth. So first of all, there was a challenge. This protocol couldn't be reproduced with conventional human iPS cells and ES cells in the beginning, but they are primed. So we thought, what about if we just use naive cells for the same protocol? And I guess it was the first and only time in my life that an experiment worked just for the first time. Yes, you just can put them in that media and do the same with the old version, with the new version, and we can make human PGCs. So the bottom line, what these experiments are showing, first of all, that the starting state of the cell really influences the differentiation outcome. And that is exciting and scary at the same time, because if I asked about the PGC only as a paradigm, I would say that we still don't know why do we must start with naive stem cells. Why do we have to do priming before we give BMP4? Why is very long priming actually bad? So you can imagine this as like you have windows of competence that open up and close down transiently. And things has to happen at the right time, at the right magnitude to be able to reach the end point. We don't understand the molecular mechanism exactly why. And what's scary about me, but what if this PGC paradigm holds up to other cell lineages? And we must face the reality that today we can reprogram cells very easily from anyone, any age, but differentiation is quite tough. We can make very few cell types and really as authentic cells that we would expect. That's not to say that's not possible, but let's say it's proven a tougher challenge than when we had thought 20 years ago. So what about like HSCs, blood stem cells, even in the mouse, for 30 years, nobody can make at the moment authentic HSC bone marrow cells. So what if the HSC is a much longer ride to get there? Maybe there we have these 20 windows of competence that have to happen at the right time, at the right space. And that is why we're not getting the cells, or we're not getting correct cells and so forth. So that is one scenario. And really you start thinking about differentiation. Typically differentiation, the way we do it in the dish is we dictate the morphogens, which ones we give, when, how much. We dictate the elasticity. We dictate if there are going to be neighboring cells or neighboring tissues or not. Now, you could argue differentiation is not really working in the dish because the cells are not that okay. But I don't think it's the case, because nowadays you could take most ES lines and IPS lines, inject them, of course this is in mouse, put it inside the embryo, put it inside the uterus, and you will get a mouse that has beautiful bone marrow cells that you can transplant over and over. So the starting cells are fine. We are the problem. We are not able, I would say, in many, many cases, to recapitulate the environment, the complexity, the sequence of events that happen inside the embryo. And really, that's why we were starting to think maybe we can somehow create embryo-like environment and use it to differentiation for human cells. But before human cells, of course, like everything in this field, we start with mouse cells. And first, before we're trying to make stem cells become embryo-like outside the uterus, we need to learn how to do it with natural embryos outside the uterus. Like in a way, if you want to go to Mars, let's try to get to the moon first, and then maybe you have better chances of making it. Or if you want to grow iPS cells, you better know first how to grow an ES cell. So first things first. So for us, the first thing was really try to grow mouse embryos outside the uterus for very long times, and then take these conditions and take mouse stem cells and ask what would happen if we would put them under these conditions. And hopefully we will get embryo-like structures, and from that we will learn and move to human stem cells. So that's the sequence of progress we have made. It's first, ex utero development of natural mouse embryos, then try to make stem cell-derived mouse embryo-like entities, which I will abbreviate now synthetic embryo models or stem cell-derived embryo models, I'll just say SEM, and then try to move and make human SEM from different stem cells. But I want to also go back to the naive and primed. As I told you, in research we were a bit also stuck, so we had another motive to do this, because as I told you, I can go between naive and primed, but if I make, let's say, a mutation in this transition, I have no readout. I cannot, if I want to know what's the differentiation outcome, I need to have an embryo-like thing coming out of these stem cells. And I don't have this sequence, so I'm a little bit experimentally stuck. But again, if I want to, as I said, we're not stuck with stem cell-derived, also with embryos, because we don't have a system where we take a post-implantation, which is like a primed stem cell state, make it an embryo. We couldn't do that in the dish. So again, we need to first learn on natural embryos, and maybe then pedal back and try to do this with stem cells. So this converges on the problem we face, which is what's called the uterine barrier in mammalians. So the uterine barrier has multiple dimensions. First, in mammalians, we can grow embryos only up to the blastocyst stage. And then, like in IVF, that's the treatment. Once you reach the blastocyst 64-cell stage, you have to put it inside the uterus, because one day later, it will no longer be able to implant inside the uterus. But the thing is, the drama of the embryo making its organs, in mouse, it happens in 5 days out of 20. It's very quickly. In humans, embryo makes its organs in between week 2 and week 6. The rest is just growth. So, and this happens just when the embryo goes inside the uterus. You have this, we'll talk about gastrulation and then organogenesis. So the embryo is very small at these stages. It is in vivo inside the abdomen. We don't control the environment. So it's really limiting for us as researchers, even those working on mouse. Another dimension of this, of matter, is actually, even in mouse, if you take an embryo, as I said, out after the blastocyst, once you take it out of the uterus, you cannot implant it in another uterus in the mouse. Once it's out, it's out. You analyze it, that's it. So in our development studies, we're very much based on studying snapshots of development. We cannot do the experiment of taking, let's say, an embryo at this stage, do CRISPR, and watch over multiple days the same embryo, what is happening to it. And as you can imagine, that's a very limiting thing. And third dimension of the uterine barrier is when you talk about humans, which of course you have all the prior, but the situation is much worse, because getting human embryos for these first month of development is technically impossible. Ethically, for the right reason, it is impossible, because usually the woman doesn't know she's pregnant. If for some reason she needs to have an abortion, it is done just by a pill. There's typically no need for surgical procedure. And even if we get samples of embryos, which we do, and people in the 60s, this is not enough to do research. To do biology on development, you need millions of embryo samples, which we are never going to have to be able to get in humans. The samples we do get are very important. They make atlases. We try really, as a field, we're really desperate. We try to do all what we can. This starts also back in the 60s with a Carnegie collection of sections. But this is never going to, I think it's only good for reference. It's never going to be good for experimentation. Again, for justified ethical reasons and technical reasons. And this is another opportunity I want to just make a side point. But also we often, when we get later discussions about how long we should grow embryo models or embryos so far, I often hear this claim say, oh, just go until day 28, because after day 28, it's much more easier to get samples from abortions. But I remind you again, abortions, you're never going to get millions of samples to do experimentation. This is good for snapshots. It is important, but that is never going to be a replacement and really a robust platform for studying human embryology. So going back to my work in the mouse, I think that there's a bigger question here also, a little bit, again, philosophical, which is, are we a little bit like frogs and zebrafish potentially, meaning that the uterus is not needed to make our shape, mammalians or in mice? Or is the uterus in mammalians really needed to create the morphogens of the embryo? In other words, can we capture the entire gastrulation outside the uterus? So gastrulation is the process of building the progenitors of organs, let's put it this way, and then can we also go to the organogenesis process outside the uterus, which is the actual making of the organs? And I remind you, this is also important for us because our end goal is to do this from ES cells and iPS cells that correspond to pre-gastrulation stages. So for us, as a protocol, we must be able to do this from pre-gastrulation. There's been really marvelous work on trying to grow mouse embryos outside the uterus. Some of it can be dated to Dorothea Rudnick over 100 years ago. And since then, I would say two people really advanced the field. Dennis New in the 60s, Patrick Tamt in the 90s. And they've built different systems. I'm just showing some examples, and they built these initial wheels. But their success was very, very limited. They could grow mouse embryos for 1.5 days at the most. So it's like from 8 to 9.5, or like from 9 to 10, different windows. But they could never capture very long periods of time. They could never start from pre-gastrulation. They could never capture the entire gastrulation. As I mentioned, it's very important for us. The efficiencies were extremely low, and most actually embryos were abnormal by default. But it was still very useful. For example, Rosa Beddington wanted to show kind of ectopic early axis or head formation. So she could transplant things and watch them grow. So for that, you don't need a very normal embryo to do that. So you can do this for, like, I want to say crude experiments, which are very, very important. But what we are after is really get efficient, normal embryogenesis over a prolonged period of time. So this is really the work who pushed this. Alejandro Aguilera, who's a very talented PhD student, who's now starting his own group at Janelia at HHMI in the US. And we started thinking, let's take some of the reagents, some of the incubators that were done in the past, and try to like improve them, re-engineer them, use new media supplements we have, and actually we were hoping for like maybe one more day at the time. So we started really re-engineering stuff, engineering, but really we end up with this ugly structure where we built this incubator here. We added this red bottle, which is very critical because it's both a humidifier and controls the flow and the pressure of the gas. We have this electronic gas regulation module, which is really a gas mixer that controls a lot of the parameters that we learned we need to control. I want to emphasize also it's very important that you make very high quality gas mixers because really it needs to be controlled. If we have like 5% missed error range, it's not good. And we have a setup, what's the pressure at each time point. I want to say that this is all published. We have Jove videos. This is available to academia for no profit. You pay the same thing I pay. And we're also working on better models because the system is a bit fragile. I want to say no offense to any British people, but like my father always said, he loves British car, but you have to buy two because one is always in the repair shop. So we're dealing with a similar case here, but we're trying to, but it works. And then we learned a lot what needs to be important. So depending on the stage you start, dynamic range of oxygen, different states need different oxygen, humidification is very important. The pressure in which the gas is pumped at different stages is important. Of course, the gas flow. Things like avoiding just putting a diaper is very critical. Because, I mean, it's not that you can work regularly, but when you're leaving it to incubate overnight, if you just leave it under the light, it is phototoxic and your efficiency will drop very much. So a lot of these kind of technicalities that add up. And the second, then, what about the media we want to grow around them? Because they have a placenta, but they're fed by diffusion. So a lot of, took rat serum, combination with human serum, HEPIS buffering, high glucose is very, very important. And if you do that, you can really get a very high efficiency over 85% from multiple backgrounds. Here we start with 7.5, which is late gastrulation. You can see in one day you have the yolk sac, you have the heartbeat, you have embryo turning, and we can reach day 11. And just to show you, it will become relevant later, that although the system is efficient, but things have to be done right. So the control I show you here, it is the optimized condition I've just introduced. And let's see what happens if you don't have the right pressure. These are embryos, but clearly they're abnormal embryos. What if you don't have enough glucose? This is what you have. What if you use the wrong serum? For example, KSR, it's a synthetic serum typically used. It's just terrible for embryos. So just shows you that parameters are very, very important, and this is also scary. It means probably there are more things we don't know. But this is exciting, but day 7.5 is late gastrulation. I told you we must start from pre-gastrulation, either 6.5 and 5.5, and there, if you do the roller culture system, they don't like it. It's just too rough for them. But what we found, we can use the same media we made, which is called EUCM, and use these IBD plates, which are typically used for human embryos. And you can see here, we can get extremely high efficiency of gastrulation. You can see this embryo growing, the growth of the extraembryoidal tissue. The red here, you see the primitive streak, you see the somites, you see the head formation, already alluding to you the advantages that we can really now start using some of this vira for imaging and so forth. So then what was really new here is really to combine the static conditions in the first part. At day 8.5, move to the roller culture conditions. And there you can really do it from day 6.5 to 11, or from 5.5 to 11. And we've done a lot of tests, really, when this is published, and just to tell you that really the embryos are very, very, very similar to natural embryos in terms of gene expression, temporal expression, single-cell RNA-seq, and a lot of tests we have done on, again, these are natural mouse embryos. So, as you notice, I intentionally don't use the words artificial uterus because it's a little bit maybe cheesy, and I have no proof that I actually have recapitulated the conditions of the uterus. But what I do think this is an incubator that does support mouse embryogenesis. I'm sure it's not the best condition, I'm sure it's not the best system, but it is a system. But I will give the credit more to the embryo in the end, that it is about self-organization. Or in other words, you don't need, let's say at least by 100%, to make morphogenesis, you don't need the uterus. You can get a lot of morphogenesis happening outside the uterus. And I want to remind you also, we're using very neutral overall conditions. So we don't put wind and BMP at different stages so we're not stimulating them. And I just want to say as a side note, before I move to the next step, that again, as I mentioned, we're not just doing this for the sake of growing embryos or embryo models. We want to make systems we can analyze and perturb. So one example of tools that we made is, I told you my experiment is really to take an embryo, mutate it, and follow it over time. So now I can do this, and we do this. So for example, you can take a lentivirus, with a GFP, or it can be a CRISPR. But you can see GFP is innocent. You put 1 microliter in the proamniotic cavity at 6.5. It just labels the entire embryo without damaging it, and we can grow it for 5 days. It's actually something we cannot do in utero. So we then move to the next stage, which is, what about embryo models? Okay, now that I have a condition, a system, I can try to think which stem cells I want to put in there, and will they make it? And there are a lot of embryo models, very beautiful work from different groups, from Nicola Rivron, from Zernike Getz, from Alfonso Martinez Arias, that made different models. But I would say the field was a little bit stuck in a way, because in one way, the blastoids so far, if you put them inside the uterus, which you can do for a blastocyst, they don't develop. That is not to say that's not going to be possible. And if you look at post-implantation here, embryo models, there was no way to evaluate whether they would develop further, because as I told you, even a natural embryo at these later stages, you cannot put it inside a uterus. So you cannot expect the synthetic embryo models to outcompete natural embryo. The other thing, which I will just show in a second, I would want to say that an embryo is not only about being, it's not only about looking at a static image, it's about being able to become the next stage. Or as my students sarcastically say at the products, it's like if you're an embryo, you're only as good as how you're going to look on the next day. So, and actually I could tell you that these structures, which look very nice, equivalent today maybe 6, they actually don't develop further. So I'll just jump forward. And if we use an optimized protocol that we know now it works for synthetic embryo models, if you take these structures here, in the same protocol, they will just give you empty yolk sac. And the reason is that they were made originally in this bad KSR serum that I told you about. So they can reach this stage, which looks normal, but they don't have a developmental potential like if you use other conditions. So which is a very tricky business. So we wanted to do this from stem cells, and we wanted to do it differently. And here from our work on human naive pluripotency, we got a clue. So if you look here, the early mouse embryo has 3 compartments. It has the post-implantation one. You have the embryo proper, the embryo itself, the yolk sac surrounding it, and the trophoblast. And while our work on human cells, and from also other groups, we noticed, unlike the mouse, it's very easy for them to become trophoblasts that make the placenta and primitive endoderm that make yolk sac. Very easy, as in you don't have to give overexpression of transgenes and genetically modify them. You can do it efficiently, sufficiently only with cytokines. So that kind of told us maybe we'll do it in mouse, maybe with genes, but let's start everything from naive pluripotent cells. And also we had in mind, because if I'm going to try to take now placental cells from embryos, when this is going to get to humans, we don't have access to authentic materials. For example, there are no really, I would say, valid extra-embryonic primitive endoderm cells at the moment that we can grow stably in the dish in humans. So taking you back to the mouse, the idea is to start with naive cells and split them into three. One fraction is untouched, which will make the embryo proper. One touch will be nudged for 24 hours to become trophoblast of the placenta. One fraction will be nudged to become primitive endoderm of the yolk sac. After 24 hours, put everything together, and then as soon as possible, get them out on the same conditions, same device, and see what happens. So just one thing we had to do, for example, we had to have a protocol to make very fast trophoblast differentiation. We want things to move very fast. When, for example, we modified this, is that we add this LPA, which is an inhibitor for Hippo pathway, but what you can see on the left versus on the right, you can see that the peaks of the trophoblast appear much early and much higher. So we had to play around with how much fast we can push the cells. And I want to go back, just here is a marker for trophoblast, and here the starting cells are naive versus primed cells, and you can see this is a quality that only naive cells can really convert easily to trophoblast. So really, again, going back to the importance of the starting material. These are the 2 points I want to make. So in the end, we optimize the conditions, which genes we should use, which cytokines. We do the nudging, I would call it, aggregate them, and modify the technicalities, shake them. Again, everything is videotaped with the paper, doing the experiments to really try to make things as possible easy. We start with 25 cells. The first 3 days, not much is happening. You would think just a ball of cells is growing. But actually, at day 4, you start seeing a polarity, that this is very similar to day 5.5 in the mouse. You have the extra embryonic ectoderm is going to become the placenta later on, and this is the embryo on the other side. 1 day later, you can see the EXE growing, primitive steers starting to form. Another day, you can see like amniotic fold, amniotic cavity, very similar to a mouse natural embryo. And day 7 is boom, you get the yolk sac expanding. It's very dramatic, and clearly you can see you have the ectoplacental cone, and you can see the embryo-like structure inside of it, very, very similar to a natural embryo. And we can reach so far day 8.5 in mouse development. It works with about 1% to 0.5% efficiency. It works with 40% of the ES lines from different background. We don't know why. We don't have a pattern yet, but still it works in several lines at a consistent efficiency. And again, if you look at the structures now at these stages, day 8.5 is post-gastrulation, and already organogenesis starts. You will see the neural folds. You'll see the heartbeat. You can see also some of the valves and the sections. You have the allantois, which is going to become the umbilical cord. And also, if you look at the yolk sac itself, so for example, the first blood cells appear in the yolk sac. And you can see here these clouds appearing, which are the first blood islets, and we've characterized them. Actually, we've now transplanted them. And they just do like the natural blood cells at day 8.5. We looked at germ cells, for example. Sorry, it does not move. Germ cells are formed in the posterior part of the embryo, and here we use a reporter. You can see the germ cells really appearing in the posterior part, day 5, and you can see them here migrating towards the gonads at day 8.5, which is an expected phenomenon. So to summarize, this is what it looks for an embryo model, and this is what it looks a natural embryo. You can see that the great similarity, the expression pattern and sequence is very, very similar. And in the end, we get a structure that clearly you have an organization of an embryo. You have the neural fold, you have the heart, you have the tail bud, you have the neural tube. But I want to emphasize an important point here. This is actually not the best embryo model I'm showing you that we got, but for all of them, you will find some defects. For example, you can see here the neural tube is too much curvature. If you look at the neural fold, it's a little bit bigger and not tilted in the right direction. If you look at the heart, it's a little bit bigger. So maybe that is, I just want to say that that they're not identical at the moment, and that's why probably they die at day 8.5. There's also beautiful work done by Zernica Getz. We are authors on this paper just because we provided the system, which reached some of similar conclusions on this, but they didn't start everything in this paper from naive stem cells, which is very important for us. Again, single-cell RNA-seq, these are the in utero gold standard. These are different embryo models from the mouse we did, and you can see that the map really looks very, very similar. We can quantify the similarity of about 95% which is very, very high of the quality of the cells and they express. So we have a lot of questions there. Why the efficiency is low? How can we reach later days? How can we also for natural embryos improve this? Can we do it from other species like rabbits? A lot of questions. But we started in parallel. We're going to the third question, which is, can we now do this and transfer this to the human stem cells? So really our goal was to really reach up to day 14, which is early post-implantation human models. And this is the work of very talented students, Bernardo Oldak, Emily Walshes, and Vlad Bondarek, a postdoc in my lab. And just to get everyone on the same page, I'll just show you these YouTube movies just to understand, and you already see how different a human embryo looks like than the mouse. So first, this is the blastocyst. It implants very deep in the humans. And also, you're not going to see this egg cylinder shape go down. The pluripotent cells here, the blue, they stay flat. And here is the hypoblast. This is called the bilaminar disc. So there's no egg cylinder. It doesn't go down. It stays like this. This is the yolk sac. And the rooftop of this thing is the amnion. Now we look from the top, you can see why it's called a disc. It has a disc-shaped, and this is the epiblast. The embryo proper is going to be on top, and now we don't see the yolk sac, which is underneath. And then cells, the posterior cells start making the markers, setting them up to start gastrulation. So that is one part. But then if you go to the next stage, there are differences. If you look at the structures here, you see the bilaminar disc, and suddenly appears another tissue, which we don't have in the mouse, which is the extra-embryonic mesoderm. As you saw in the video, suddenly it blacks and it appears because we have no idea where it's coming from, just to show you how much we don't know about human embryos. But this tissue is also very important because it starts actually shaping other tissues, which is, let's see it now. Sorry, it's the next movie. It expands, it makes this cavity, which is the chorionic cavity, and you can see the yolk sac actually is shrinking. It becomes a secondary yolk sac. Some parts of it are the remnants and other compartments. And here I just want to emphasize how difference is, a mouse is great, a mouse is not a human. I would even say even in the end, monkey is going to turn out not a human. So it's very different. Until the blastocyst is very similar, after that, different game. And you can see here also these lacuna, which are cavities more found in the trophoblast that fill up with blood and important for tissue exchange. So to tackle this, we asked, so this is the structure we're going to try to mimic. It has 4 cell types, not 3 like in the mouse. The primitive endoderm for the yolk sac, the extra-embryonic mesoderm, and the trophoblast. And we wanted to start with naive cells. And luckily for us, as I mentioned, we already solved the initial problem. We already had naive pluripotent cells in humans. So we could do this. And we want to ask, how do we push them to become these cells? And typically in these protocols, as I show you, it's very important because you're mixing different cells. You want your cells that you think are trophoblast to be 100% trophoblast and not to have junk in there. So it's very importantly to push the cells quickly into a cell type, and that's what we wanted to figure out. So in the mouse, when they make the PRE, we express GATA4, for example, you can see in 48 hours by fax, you get these PDGF receptor alpha, which means their primitive endoderm very, very high up. So really, they just move very, very quickly. And in the humans, we had to test many, many conditions to test how do we make these, let's say, fractions become very quickly. Do we need transgenes? Which cytokines? And that is the point. And actually, when we were trying to do the primitive endoderm, we were in for a surprise. As I told you, in humans, we can do it just with a simple media, no need for transgenes. You can see here 75% convert. But also, we noticed that with the same induction, we're getting 2 populations. We're getting both the primitive endoderm, you can see here the SOX17 cells, and we're also getting the extra-embryonic mesoderm, which is these BST2 red cells. And you can see there's no overlap. So we're getting with the same induction both populations. What about trophoblast? We've tested a lot of prototrophoblasts that exist, that were published before, but here there's another challenge. You need the cells also to go to the right place after you aggregate them. And in humans, trophoblasts have to go to the surrounding. So trophoblasts here are in pink or magenta. As you can see here, trophoblasts from certain protocols, they would just never sort. They would just stay as a separate clump. Perhaps they're too advanced, perhaps they're not authentic, we don't know. But we found one protocol, again, without gene modification, you could see over 70% efficiency, they will sort out to the surrounding. So after that, we moved to the experiment, of course, optimizations of the media and the shaking and the volumes. And this is what we end up with an 8-day protocol. We put 120 cells. First 2 days, not much happening. And then by day 3, you actually start seeing that in 1% of the structures, again, you see compartments and cavities. And of course, we validate this. I'm showing you the phase image, not the staining, but we validate these compartments based on the stainings. So this is what it looks like typically. If you look just at the section, the trophoblast, the pink, are in the surrounding. You have the epiblast in blue. The lower part is the epiblast. This is the amnion on the top. And you can see the interface with the hypoblast, the thick part of the yolk sac. And so this is the bilaminar disc. I would say this is the most essential defining feature of a post-implantation embryo, without which no organogenesis is ever going to happen. And this is the yolk sac. Sorry? Oh, I'm sorry about that. So yeah, the blue is the epiblast, the yellow is the yolk sac, and if you see, there's another small cavity under the yellow, and this is the chorionic cavity. So you have like 3 different sacs on top of each other, all surrounded by the trophoblast, and that is the hallmark. You know, easiest is a movie. Here you see the movie of equivalent day 8 or day 14. The trophoblasts are on the outside, and now you're going to see now the yellow is the yolk sac. The thick yellow is the hypoblast. On top of it, the blue, the part, this is the epiblast, the embryo organogenesis is going to happen there. And the rooftop of the blue is the amnion that you have in there. And this is, of course, this is not an animation, this is segmentation, this is the actual stainings. We go through different sections, and thus we basically build up what this looks like. And really, again, because in the end, when we say embryo models, you have also work, it's a model of an embryo. And an embryo is about structure and having in the right orientation and configuration, which we were very happy to achieve here. Not to say that they're identical, not to say that they are perfect, they're not. But we're getting much more than before. And this is just if you take a look at the section, and if you open a typical scheme from an atlas, you can start seeing that really, you can see that the very striking similarity between the two that you get, that you don't need a lot of imagination to see this. We can dig a bit more in the compartments and see processes. For example, this is what starts with the epiblast, and it's going to make the lumen of the amnion. Okay, so this is the rooftop. You're going to see now when we dig deep, we're going to just start going inside and see the lumen opening up. So we're going to go now inside. It's a closed ball, and then here's the early lumen inside of this. And this is a very, very early lumen. So we went deeper to characterize them. I'll just walk you a couple of things. You can see that the lumen is forming. If you look at the different stainings, you can see if you start with the blue, very small lumen, it grows every day, and the epiblasts grow much more and more. If you look at the cavity too, you can see the lumen for the green there with the arrows. So the proteins that are important making a lumen are really expressed there. If we then ask, well, did we still go through a blastocyst during this aggregation? What happened? What stage? And we don't see any evidence of going through a blastocyst. So as you can see in the top, the blastocyst there on the blastoid, it has a blastoid cell cavity. But we don't go through it. The earliest identifiable structures, which you will see here in E, this is clearly already becoming a post-implantation structure. This is also similar in mouse. So somehow, I would say as if when we're starting from a deconstructed blastocyst. And as it's the cells coming together, these first 3 days are basically start sorting directly into post-implantation stage. If we look about the amnion, we didn't use any human embryo tissue that we made here. Here are atlases from the 60s of the Carnegie collection that are very useful. You can see 2 things, how the amnion epithelium is thinner. So the upper blue is always thinner. And you can see they have different gene expression. So TFAP2A in red is only expressed in the amnion and not in the epiblast. And this really matches results from Fred Lanner. I was here on the monkey. And here I want to highlight also that the monkey and the non-human primate research, although we always have to take things with, maybe it doesn't hold true, but as I said, we are desperate. So we're trying to look for whatever useful thing with great caution that we can never have, and for the right reason, the perfect control. We can't just go in, like in mice, dissect day 14 embryos and start comparing them side by side. And this is just, again, rendering of looking at the amnion cells, just the movie, and looking at the compartment. The amnion is on top. and the epiblast, the blue, is below. And I want to show you, when we look at the top, you can really see that it looks like a disc from the top, like a donut, which is very, very different than what you see from when you're working with the mouse. Just showing like this, and you get the outline of a disc. What about germ cells? We can see that they're also expressed, the rare cells in the epiblast or the amnion. It's a big question. We didn't get into that, I must say. But to see that they are there, they're specified. What about the yolk sac? You can see here the yolk sac, this is the cavity. And similar to in vivo, there you have some of the epithelium is very columnar, and some of the epithelium on the side, the parietal epithelium, it's very, very thin. So let's look at the movie of the embryo model. You see the cavity forming into the yolk sac, and you dig deeper, you see here the visceral endoderm, yolk sac, columnar epithelium, you get here parietal epithelium. So really we're getting fine details so it's what I call dynamism, which means that once the cells are assembled, it's not enough. To get a picture is not enough. You need to see that you get sophistication and growth, not on size, it's about in the stages. So that was very important for us, and we had a lot of different examples. What about making what's called the first axis of differentiation? We also see that. You can see the brachyury, which is a marker of the posterior epiblast, the pink at the back. You can see actually the stalk, which is a structure, again, not in mouse, which is an extracellular cells, the ECM cells, hold the epiblast to the trophoblast. And the green that you see is a very important structure called an AVE. These green cells are vesicles of molecules that start dictating the gradients of morphogens. So again, this is called the anterior-posterior axis, where you have the pink in the back, in the epiblast, and the green just on the other side beneath the epiblast. What about another structure, the chorionic cavity? So these extra embryonic mesoderm cells start migrating and make this cavity. As you can see, they have no, with the white, they are no SOX17, and they have GATA6 low. It's a bit lower than the primitive endoderm. And again, this is exactly what was observed in monkeys. So again, also teaching us that we're really getting fine details of gene expression patterns in the model. If you look at the extraembryonic mesoderm cells here, we saw that they really migrate to the right position. So these are the red Vim-positive cells. You can see they're between the trophoblast and between the yellow yolk sac. So really not going inside the yolk sac, not going outside, they can really start expanding and make the chorionic cavity there. The last compartment we look at is the trophoblast. And there, the secondary yolk sac, we also see that. This is again the Carnegie. You can see the extraembryonic mesoderm starts growing and you get these cavities open. You see some trapped SOX17 cells here. This is the remnants of the primary yolk sac. You can see the secondary yolk sac is actually smaller and it is developing at this stage. What about the trophoblast? So we see a lot of phenomena. We can start seeing that they start secreting hormones like beta-HCG, and if you put it on a stick, you get a positive band of pregnancy. That doesn't mean that's a real embryo. It just means that it secretes a hormone. And the other thing, cells fuse the syncytium, and you can see here a cell type. This is one membrane. The red below, if you can see it, and then you can see the green there, 3 nuclei, or 4 actually, in the same cell. So you're getting what's called a syncytium, like multiple cells in one, multiple nuclei in one cells. Other features is what you see on the right, when I mentioned the lacuna. These are structures, again, we don't see in the mouse. In humans, they get filled with blood, and they're within the syncytium. And if you look in 2D, you can see the sections there. These are the open cavities. But again, a movie is much easier. Here we're looking at day 14, beta-HCG in green, and you see this window. You're like peeking inside a window. And this is the lacuna that develops in the trophoblast compartment on the outer. And this is now you're looking at the 2D section. So just going like this. Again, just showing you that microscopy is very, very critical for us here, particularly in humans also, models. The trophoblast, unlike in the mouse, it surrounds the entire thing, and trophoblast can be thick. So it's not easy to see phase contrast based on the eye unless you really, really experience, and it's a challenge, but we'll leave that for the experts. I want to skip this. We did single-cell RNA-seq analysis to really make sure that the cells are there. But really what was really critical for us is really, not because he's here, but beautiful effort from Fredrik Lanner and Sophie Petropoulos, who are collaborating with us, who made on the left, you'll see it's basically a compendium. They took almost all data sets that somehow were scattered on human embryos from samples, made a reference map, and then we took on the right our different cell types, with the trophoblast, the yolk sac, and we can see that the cells are really almost falling where they're supposed to fall. And I mean, again, it has a lot of limitations, cross different platform differences, but at least it's very, very encouraging. And also we don't see still naive pluripotent cells or some other cell types. So things are looking that they're very, very similar in their way. So I finished by saying that a lot of conclusions can be made from this, whether about pluripotent cells are in a way de facto totipotent. They still have the potential to make the trophoblast and so forth. They can both start making models up to the gastrulation stages. In mouse, we actually can go through gastrulation and start organogenesis, which we haven't reached in humans. And of course, there's a lot of things to talk about this. But really the big question, which is going to be discussed later, I like this, the Magritte painting, which is this a pipe? And we made our kind of fun take on it just to say, this is not a mouse embryo. And I'm not making a declaration that this is not a mouse embryo. I'm actually kind of maybe being sarcastic in a way. Is it? Is it not? I don't know. But that's where we are. And just finish by saying that I want to remind you that why we're doing it in humans, because a lot of reasons. The first parts of pregnancy are very critical. A lot of abortions, a lot of developmental defects happen during these windows. We can't study them yet. I want to say the efficiency is very low. 1% is too low, but I believe we can go higher. But still, for example, also for drug testing, you cannot test drugs if they're dangerous to babies on women. So some new drugs that can never be given to women. But maybe if we think, which we proved in mice, if you have an embryo model outside, if you give it here, valproic acid is a known drug that causes brain defects. And you can see here, we can just add it to the mouse embryos, and within 24 hours, you see in the white arrow that really the neural tube doesn't close. So really, we can see these effects that we know. And maybe one day we'd imagine that this can be kind of an initial test for drugs, and maybe if they pass this test, maybe the FDA will allow testing, or for pharma, I don't know yet, but these are ideas floating around. And the last idea, which is first, as I told you, we can test genetics, which genes are important for which tissues, because we start with stem cells, we can mutate them with CRISPR, and we can go further. But the question is, since basically I'm showing you that differentiation, this is a differentiation protocol that happens on its own. Or in other words, naive pluripotent cells have the potential. We just create the environment to unleash that thing, and they start cross-talking, secreting to each other. And are any of the cells there useful for transplantation? And we already see in the mouse that the quality of the cells we are getting is much higher than what we do with 2D differentiation. So we can imagine, and this replaces the ethics, and what is the scenario here? Maybe, let's imagine a patient who has leukemia, cannot find a bone marrow donor, he's about to die. Can he have the rights? Is it ethical to take a skin biopsy from him, make iPS cells, make them naive, make an embryo model to day 35, where at that time you have blood stem cells in the liver, which were known to be the best cells actually for transplantation. Justified, not, worth it or not, I don't know. And I finish by thanking the team members I mentioned during the work who worked very hard, our collaborators, Fredrik and Sophie, and also Vincent Pask from Belgium, different funding agencies, a lot of conflicts of interest that I must declare. And last, we're always looking for very talented students and postdoc, and if anybody is interested in joining my lab, please talk to me. Thank you, and happy to take questions.
Thank you, Dr. Hanna. Thank you for an outstanding presentation. So now we have some time for questions in the audience. Erna?
Hello. Thank you so much for an interesting talk, Jacob. Heidi Benson, University of Oslo. So you were saying, well, we all know that in several countries we draw a line at day 14. And this has been due to the primitive streak appearance, etc. You've shown that that line is maybe not correct. And you're also saying that after day 28, we can't really depend on aborted tissue because we would need millions. And so where would you draw the line, and why? Would you draw it at 40 days due to organogenesis or somewhere else?
Yeah. So I just want to make sure that I didn't say to change the line or not the line, because we're still talking about an embryo model or synthetic embryo model, and it's a big question: when should it become an insert like an embryo? In the end, that is the question, because I think overall, maybe it's simplistic, but the general, we have been discussing, and there's a lot of regulations on embryo research. It's different between countries. Israel doesn't have a 14-day limit. Iceland, I believe, the Nordic countries do have it. The question in the end, when does an embryo model become an embryo? And I think then the regulation will be the same. But of course, it's a question when this definition of when did it become an embryo is a very complex one. So that's why I don't know. I have my opinions about certain things. I would say I'm very much an advocate of the policy, what I call fingers on the pulse. I think there's no alarm to sound. I think we are very far from very advanced stages. Even day 28, it's going to need a lot of work. Efficiency is low. We already see the differences. So that means actually we have time to discuss and think about it, which is, I think that is why we are here, is just to not only actually also to express my opinion, but actually also we often as scientists, so most often we want to hear back feedback, and maybe there are points we didn't think about, and hear from religion, from society, patient advocates. So I don't know. I mean, for us, we have ethical approvals to reach day 28 with embryo models from iPS cells that were donated specifically for this experiment. We're looking forward to this discussion. I want to say that how things shape up. But I would want to just finish two things. So 2 points. As I mentioned a little bit, when I think of ethics, I don't think of it as in a vacuum, meaning it's just to say, is it ethical, or what's an embryo or not an embryo? There's always going to be an ethical price. I think of it in this scenario of this leukemia patient or an older woman who doesn't have eggs, and it's important for her to have her own eggs. When is it justified, and the cost is the ethics? The second point, which we can consider, we're actually showing this experimentally now, we have the option to do developmentally restricted stem cells, meaning delete a gene or two, then you don't have a heart, you have no neural tissues. You absolutely do not need these tissues to make blood stem cells or eggs that I've just mentioned. And a lot of rabbis that I've talked to and ethicists, they say this can never be considered an embryo because it can never be born, it will never have any cognition or things like that. So I just want to say that also, science might have a solution to the concern. So it's very important to go through the process and learn what are the dangers or what we don't want, and then maybe actually we can bypass it by experiments or by other way. So the process is very important, and not to kind of rush and shut the door and just simply last sentence, you don't ban nuclear physics because somebody can make a nuclear bomb. And trust me, a nuclear bomb is more dangerous than all what I've presented right now.
Thank you. My name is Erna Magnúsdóttir. So I have more of a just straight biology question. It's so nice to see that you're talking about the germline, not surprisingly.
Oh, it's nice to meet you. Now I know your papers, but I never met you.
But do you think, so often somehow the germline gets overlooked in these studies?
Not in my case, yes.
No, exactly. Do you think, because you showed, and I believe, I haven't read your paper thoroughly, but you say the sum of the embryos at least for the germ cells, but do you think, so first of all, it's always is it somehow harder to make the germline, or just the inefficiency of kind of getting germ cells just reflect kind of the other problems, kind of the shape of the embryo is always...
No, it's a great question. I think it comes, because, germ cells really start to appear at this very later points that we are at, like day 13, day 14, as you start seeing them. So because that efficiency is low, but when we do see them quite often in these structures, in that aspect. But I was, had a long discussion with Azim, as you know, in the field. There's a big discussion about are the PGCs from the epiblast? Is it from the amnion? I'm very careful because there are a lot of, I think we need a bit, maybe 4 more days to really know for sure, because maybe there is like a sporadic emergence in the amnion, but that is not the real source. Actually, the real source of the posterior epiblast. So I wanted to be very careful about it. We need just 5 more days, I think, and that's one of the things that we're really dying to look at. It would be also much easier if we can increase the efficiency. I think we can.
Hi. I had one question. It's a practical question. You mentioned that you can perturb gene expression with a lentivirus in the mouse?
Yes, yes. I mean, a lentivirus expressing like a CRISPR, basically, like a guide RNA.
Okay. I was wondering if you can use something like siRNAs or something with embryos, because that's something you usually need to transfect into T cells. So, would you transfect them into embryo? How does that work exactly?
That's an excellent question. So I'll link it to what I called about developmentally restricted stem cells. What we do in the mouse, for example, there's a gene called LIM1, without which you have no cranioneural tissues at all. So we take the natural embryo or the embryo model. We can protocol transfection. The embryos, they really survive transfections, electric shock. So we put the guide for that gene very early on, we electroporate it, and that tissue will not develop. So yes, that's why I'm saying we have to be patient. There are going to be technologies helping us also in the ethical question, but also if you're studying basic research, as I mentioned, it's not about the titles of growing embryo models. In the end, it's an experimental platform. We must bring it to an experimental platform, and the method you mentioned is also looking quite promising.
Thank you very much. So for the sake of time, we thank Dr. Hanna.
Thank you.
Thank you. So our next speaker is Dr. Fredrik Lanner. He is a principal investigator at the Karolinska Institutet in Stockholm in Sweden. And Fredrik is studying by what means totipotency and pluripotency are established in the early human embryo, and we expect to hear more about that. Please, Mr. Lanner.
Great. Thank you so much for the introduction, and thank you so much for the invitation to come to Iceland. Whoa, it really goes high. I really enjoyed the discussions yesterday, and I hope we can have more exciting discussions today. So the title of my talk is Using Stem Cell Models and Extended Cultures to Understand Human Embryo Development. And I thought I'll start with this. Let me now see if I can get the pointer going here. Oh. Is it possible to turn down the lights? No? I wanted to start with this picture because it's still one of the favorite pictures in my lab. This is a embryo culture to day 14. It's not a human embryo, it's a non-human primate embryo. So it's been taken from the blastocyst stage and then artificially implanted just on plastic and allowed to grow for another 7 days to day 17. And then we've done a cross-section and stained for markers. And I don't really need to introduce this anymore after Jacob's fantastic talk, but I just quickly go through. So in the outer here we have the trophectoderm cells in blue, which will give rise to the placenta. Moving in, we have these really peculiar green cells, the sort of cells we don't find in the mouse, the extraembryonic mesenchyme cells, which will also give rise to the stalk region here. Next to the stalk here, we have some more blue cells. This is the forming yolk sac, or the future yolk sac. And then moving up to the pink cells, that's the magical embryonic cells. So it's only these cells will give rise to the actual body of the monkey. And they have not yet started to specialize. They are like classical human embryonic stem cells. But there are probably a few cells here next to the stalk that are just starting to delaminate and initiating gastrulation, the first step to sort of on their route to specialize into to specialized cell types. Above the epiblast, or the embryonic cells, we see another cavity, and this is the amniotic cavity forming, and it's these amniotic cells that will sort of enwrap and keep the embryo in the amniotic fluid during the pregnancy. And it has felt that we've been talking about the 14-day rule, and we might talk more about that today. And I just wanted to sort of show that at 14 days, like the embryonic compartment really hasn't matured at all. It's what we're doing in the first week is just preparing for the support system for the embryo to develop. So if we want to learn anything more about how the embryo develops, we really need to go beyond 14 days. So why would we really like to study this? We've already heard from Jacob several sort of reasons, and I'll just start with a very personal reason. This is me and my wife 15 years ago, one of the best days in our life. We just got married out in beautiful Stockholm archipelago. What this picture doesn't show is that in the years before, we've really struggled to have our own children. We have undergone several rounds of IVF treatments, several that were unsuccessful, and nowadays IVF is quite common, so you don't think about how much work and how mentally tough it is especially for the woman, right? You need to give hormone stimulations, and then you go in and aspirate with a needle, take out the oocytes, fertilize those in vitro, wait for a few days and hope that there is some embryos developing, and then the precious ones that have developed transferred back, and then there's another 3 weeks or so of nervous waiting to see if you're pregnant. And we went through several cycles of this for a few years before we finally were pregnant. And that was all good until week 18, when my wife had a bleeding, and we went into the doctor, and then the amniotic fluid had leaked out. There was a rupture of the membranes. Nothing you can do. The embryo was still fine. We could see the heartbeat. Over the night until the day after, the heartbeat stopped, and there was nothing to do. As a couple, this is, of course, extremely devastating, right? We've been struggling for quite some time. And also as a developmental biologist, I just defended my thesis in vascular biology. I know that we didn't really know the reason, either to our infertility problems or why this miscarriage. And the doctors, of course, could give no answers either. So it was just, this is the way it is. And I think this has really influenced my decisions of my research. So after that, I moved to Toronto, and joined a lab that is really pioneering in early mouse developmental biology. So we started studying mouse development, and it went well. We figured out the mechanisms that control how the yolk sac cells are specified from the embryonic cells, and a little bit how the trophectoderm cells are specified. But what was disturbing, though, in a way, is that when we try to transfer that knowledge over to human or other mammals, that those mechanisms did not hold up. They were very specific for the mouse. They don't hold up in the bovine. They don't hold up in human. So after 4 years in Toronto, I decided to move back to Karolinska Institute and try and study human embryos. So we started, at the time, single-cell sequencing was really sort of a new upcoming technology, and we were fortunate enough to collaborate with Richard Sandberg, who sort of developed this technique, and we could map human development on a gene level, which out of the 20,000 genes are expressed as the embryo developed during the first week, and how do the first 3 cell types specify. I won't go into the details of this, but I just wanted to highlight here that we ran into a roadblock here at 7 days when the embryo is supposed to implant. We can't really study that process beyond at the time. But this really changed around 2014, 2016, when multiple labs showed that you can actually culture mouse blastocysts and human blastocysts beyond the implantation in surprisingly simple ways, just on plastic or some sort of supporting extracellular matrix. So you can now, for the first time, go in and study beyond 7 days and starting to approach the 14-day limit in human. In primates, it's then been shown that you can go beyond 14 days, you can reach gastrulation, and you could probably go even further to get sort of towards neural tissue, neural tube starting to form in culturing of primate embryos. So this was my sort of introduction to my talk and the field. So what I want to do now is I want to exemplify this with one research project that we've completed and is published already, related to culture of human embryos. And then I want to go into our current work on establishing references that we can use to validate stem cell-based models and go a little bit into how good these stem cell models are. So the project I wanted to talk about today was really not initiated by me or my lab. It was initiated by another professor at the Karolinska Institute, Ken Chen. He spent his whole career, or a large portion of his career, studying how the heart is formed. And he identified a function of a transcription factor called islet-1, and that is really critical for normal function or formation of the heart in mice. The big question, though, that always been nagging him is that when you look in big patient cohorts for mutations that are associated with congenital heart disease, there's no mutation. So I think there's one report of a mutation in this gene, which really indicates that islet may not be important in human cardiac development, or there might be other reasons why this hasn't been found. So he decided to, let's move from the mouse model. You cannot knock out these genes and look in human embryos, but what you can do nowadays is to do this in primates. It's not every lab that can do this, so the way to do this was to collaborate with the primate facility in Kunming in China. Really fantastic people. And to use the genome editing CRISPR to knock out the gene islet in primates and then see how does impact cardiac formation. So this was done. CRISPR was, or the gene was targeted in the zygote. Normal embryos and the targeted embryos were cultured to the blastocyst stage. No differences were there. Then they were transferred back to pseudopregnant primate mothers, and to their surprise, they couldn't study any malformations of the heart when they targeted this gene, because the embryos didn't develop beyond implantation, or they couldn't detect pregnancy after transferring the embryos. So this indicates that there's a really early phenotype, perhaps even before the heart forms in primates. And this is where me and Ken started to talk about how to tackle this. We realized that it's going to be impossible to try and look in vivo in a primate animal and not a primate model. So instead we decided to go in vitro. And here a really talented postdoc, Ran Yang, did cultures now of primate blastocysts into post-implantation stages. And we did single-cell transcriptional profiling, and analysis was done by equally talented Alexander. And what we could see was that these implantation models worked really nicely with these primate embryos. We could see the expected cell types emerging. And the next big question was then, can we now detect islet? Is this gene turning on during this post-implantation stages? And fortunately, we could see that islet turned on in a subset of cells within the cells that we had annotated as epiblast, the embryonic cells. If we zoom in on the embryonic cells, we could see if we look closer, we actually have undifferentiated, unspecified embryonic cells. Then we have primitive streak-like cells. We have two populations that look like actually mesoderm cells are gone through gastrulation. And then we have cells that have signatures of amnion, and this is where we saw islet expressed. At this time, we really didn't have a good reference for what genes should be expressed in the forming amnion in the human. There were some insights from primates and some data from much later amnion, sort of almost term amnion. But fortunately, at this time, there was this fantastic paper, or transformative paper, published in Nature, where they were fortunate enough to capture a very early pregnancy termination at somewhere between when they weren't expecting it to be this early, but when they looked at the actual embryo, they staged this to be somewhere between 16 and 19 days, so just in the time when human gastrulation is occurring. So they did single-cell sequencing of this, and this was really useful for us because now we could compare our cultured human embryo data with cultured non-human primate data with the human in vivo data. And the important part is that the transcriptional signatures really aligned, and most important, the amniotic cells that expressed islet really had an equivalent in the human. It also expressed islet. So we're convinced that it's expressed in the cells that form the amnion. So if we go into and look at morphology, we wanted to sort of get a sort of sanitist check that these embryos actually organize in something that resemble an embryo. And now these structures are familiar to you. We have the pluripotent epiblast here, we have the amniotic cavity, we have in green the extraembryon mesenchyme cells, and then we have the future placenta cells in blue. And if we zoom into the amniotic cavity, we can see that although these stainings are really challenging and we don't have that many embryos that have been cultured, but we were really happy to see that also on a protein level we could see that this islet was expressed in amnion cells. So what is the function here? Why don't these embryos develop further? So we then took knockout blastocysts and cultured them and compared to normal blastocysts. And morphologically, we really couldn't disentangle these two, so we performed single-cell transcriptional profiling on the mutants also. And we were expecting to see a lack of amniotic tissue, but what we saw was that the amniotic cells were forming just fine in these red mutants here, but we were lacking primitive streak and mesoderm cells. And this is a bit confusing because these cells don't express islet, so this indicates that there must be something that amnion is signaling to the epiblast to induce streak and mesoderm. So we dive deeper into the amnion in the... Oh yeah, this is also just to show that the gene signatures of genes associated with streak and mesoderm is lacking in the knockouts. We then zoomed in on the actual amnion in the knockout embryos, and what we could see is that, of course, islet is downregulated, but we also saw a really key signaling component, BMP4, being severely downregulated. So, and it's known from mouse biology that BMP is one of the signals that initiates the epiblast to start gastrulate. In the mouse, that signal comes from the extraembryonic ectoderm, which sits just above the epiblast. In the human, we don't have an extraembryonic ectoderm. Instead, amnion is sitting just above the epiblast. So we hypothesized that the signaling center might have been adopted by the amnion, or in the mouse, it might have been adopted by the extraembryonic ectoderm, but that the BMP signal is coming from the amnion initiating gastrulation. And this BMP is driven by islet. So we wanted to test this, but now we realize that even these embryo models or these cultured embryos are too complex to try and sort of tease this apart. And we also wanted to move this back into the human to see if there's a human relevance. So we moved into stem cell-based embryo models, and a specific model that was established by Yangping Fu in Michigan, where he could aggregate human embryonic stem cells and induce a cavity formation, amniotic cavity formation, where the top cells here are the amnion cells and the lower cells are the epiblast that are just about to start delaminating and gastrulating here. So we thought this is a perfect model for us to look at this function. So we compared normal embryonic stem cells and embryonic stem cells where we've taken out islet. And in the normal embryos, of course, we can see islet in the upper amnion compartment, and you can see gastrulation then with brachyuria and mixel in the delaminating cells. In the knockout cells, we of course don't have islet because we've taken it out, and you also don't see this morphological delamination, and you don't see the same robust induction of mixel and brachyuria 1. In another model, we've been able to reintroduce islet with synthetic RNA to rescue the phenotype. We've also been rescuing it by adding BMP4 exogenous. So we think this sort of supports this model that would now come from. We're trying to study cardiac development, realizing that islet is expressed in the amnion, and the function in amnion is to induce BMP signaling. And we're suggesting that amnion is a signaling center to initiate the embryonic cells to start gastrulate through BMP. So this is one use of extended cultures up to day 14 and using embryo models to sort of assist in studies. The second part of my talk I would like to sort of... Oh yeah, the conclusion I've gone through already. I would like to talk about is building references to validate these embryonic models that we are establishing. And what's my time? Is it how much more time? Yeah. Yeah, okay. I started with, yeah. 5 minutes. Yeah, good. So Jacob already mentioned this. So we've been spending actually quite some time on establishing a reference that we can use in the community. So we've assembled our own data with other data to start and sort of stitch together a map or an atlas of how cell lineages develop in the human. And these are the annotations. We now have a map from zygote, 8-cell stage, morula, up to the blastocyst stage, where you start forming the trophectoderm cells. After implantation, you get cytotrophoblast, extravillus, syncytial trophoblast. In the inner cell mass, you have a specification of the hypoblast and the yolk sac. We have the epiblast that goes from a pre-implantation to post-implantation epiblast stage. We have amnion formation, primitive streak, mesoderm, and then these peculiar extraembryonic mesenchyme cells. So we have genetic or gene references for, or fingerprints for these different cell types. People have been integrating a lot of data for quite some time. What we wanted to do was to have a stable map which wouldn't change its shape if we take Jacob's data or someone else's data and add it on top. So we could add multiple data and see where do they relate to this map without changing the map. And without going into the details of that, I'll just show some sort of validation that this is working. So we then took previously published data that were not used to build this map, and we integrated it onto the map or projected it to the map. And this is in vivo data or embryo data from the earlier stage, as we can see they're mapping to the early stage of the map. We then took data from the blastocyst stage, where we have reference, or where there's epiblast, primitive endoderm, trophectoderm, and that's sitting in the right position. And then blastocysts that have been cultured in post-implantation models. We can see that especially the trophectoderm compartment is really maturing forward. And if we take established stem cell lines, that naive stem cells that Jacob talked about should be sitting in this position, and that's where they're sitting. And primed conventional stem cells sit in a more advanced stage where they should be sitting. So we're happy with how the reference is working and the projections are working. So now in the last few minutes, I'll just sort of go through some of the embryo models that we've discussed, and I'll just focus on blastoids and post-implantation models. And I just keep this Blakely et al. Data, so this is actual blastocyst. So this is how an actual blastocyst projection should look like. The very first paper that was published now 2 years ago in Nature looks like this. And you have good epiblast contribution, you have primitive endoderm contribution, but most strikingly, you can see there's a really devoid or a lack of trophectoderm signatures here. And the cells that were annotated as trophectoderm in the paper, instead are sitting on the amnion. And this is something that is not so surprising, because the amnion has quite similar transcriptional signatures. And if you don't have proper amnion references, you could easily think that these cells are trophectoderm cells. And then in addition, you have a lot of these really interesting extraembryo mesenchyme cells, and they're really interesting, but they should not be there in the blastocyst. So I think this is one of the first lessons, that even though these structures really looked like blastocysts, the trophectoderm shell was actually not a trophectoderm shell, but it was a amniotic cavity instead. This was also sort of reported by Benedict and Sosen. They used another type of stem cell called EPS cells, and they got structures that actually got trophectoderm cells, but they also had the problem of having these extraembryo mesenchyme cells that really shouldn't be there. And they also reported that although we have structures that look like blastocysts, the transcription of the gene signatures don't match perfectly with actual embryos. And that's aligned with what we see in this map also. This is another example where they also use the same type of stem cells. Now they're also getting trophectoderm cells, but they're too advanced, and you have also the epiblast cells are also too advanced, and we have some of these extraembryonic mesenchyme cells. We then move on to something that is closer to Jacob's naive cells. They're not exactly the same, but something in the same similar direction. It's starting to look better. Now we have trophectoderm, we have pre-implantation epiblast, a little bit of primitive endoderm. And then another iteration, there was a whole flurry of paper published the same year. This is from Nicholas. We heard about his really beautiful blastoids yesterday. You can see they're really hitting the mark quite nicely with the trophectoderm, the primitive endoderm, the epiblast. There's some cells where the epiblast have matured more spuriously, and then there's like 1% of these extraembryon mesenchyme cells also. So, but transcriptionally, the best data set, at least, whether that's depending on how you select the embryos or so, is another question. But is this from Yanagida et al. From Austin Smith, where you have trophectoderm, primitive endoderm, and epiblast cells. So I think this just illustrates one complexity, and especially if we want to think about regulating these by law. Like, how should we view this? All of these looks like blastocysts, but at least from a developmental biology perspective, this is not a blastocyst. This is an embryonic structure with an amniotic sac. That's something completely different. And of course, doing this kind of analysis is going to be quite difficult to then evaluate what do you actually have here on a legal basis. But I think scientifically we really need to look at this carefully, because we need to understand what we're studying. And finally, post-implantation models. These models really exploded this year and in the summer, starting with Magda Seneca-Götz's publication in Nature, where they really get nice contribution to more advanced primitive endoderm or yolk sac tissues. The epiblast cells really sit where they should, a more advanced stage. They have very nice contribution to the amnion, which you should have. Primitive streak and mesoderm signatures are emerging, and you have these extraembryonic mesenchyme cells. But what is striking is that you're, in this case, again, lacking the trophectoderm, which really should be there if you want to have a complete integrated embryo model. Then moving on to the model that Jacob just presented. Of course, I'm biased here because I'm a co-author on this and I did this analysis. But very similar to Magda, we have good contribution to this side, and now we also have contribution to the trophectoderm. We're still struggling with. We think that we should be capturing more, and there might be technical reasons where it's actually difficult to dissociate these early trophectoderm cells, and this might be also a reason why we don't capture them in Magda's data set. As a sort of flip side, or a sort of an extreme to the other end, is a recent paper where the post-implantation model was not assembled at the post-implantation stage, but it was instead started from a blastoid that was then developed further. And here we can see really good contribution to the trophectoderm compartment, but it is at the expense of the embryonic compartments instead. I was discussing this with Jacob yesterday also, whether this could to some extent be dependent on that these two models, the embryos are sort of free floating. They don't have a surface or endometrium to attach to, whereas with the blastoids there's not an endometrium, but it's an extracellular matrix where the trophectoderm cells can attach to and migrate out, and that might be why you're getting a better contribution. So that might be sort of a next step for these other models also, that to give that support that the uterine epithelium usually provides. I wanted to finish just briefly sort of listing other reasons to why we would like to use these embryos, and not just my own personal reason. Jacob did a much better sort of overview of this, but I'll go through it briefly. So I just want to sort of state, although I've criticized several of these embryo models, and there's like always drawbacks of some of them and benefits of others, we still have really good models to model the pre-implantation and the peri to post-implantation, and most probably we can sort of advance this further than gastrulation also. The most obvious that I sort of started with is also that we could learn more how the actual embryo develops, and that could sort of help us in understand causes of infertility and early miscarriages. As also mentioned, we can now potentially start thinking about using these to test for toxicology and for pharmaceutical drugs. Perhaps not just for pharmaceutical drugs, but also environmental toxins. I have a colleague that is really interested in looking at this and sort of see how do these plastics that we have around us, how do they impact early embryo development. And then we heard about cell therapy or organ replacement, and I think here we have two sort of avenues. We can both learn how to better instruct the cells stepwise without building complete embryos. But I think it's also an interesting concept that Jacob brings up, whether we should actually harness the sort of intrinsic power of the embryo to sort of organize these structures themselves. But of course, we're getting into more complex ethical discussions then. And then finally, I think, from my perspective, it's also a way to replace animal experiments, especially if we're going into non-human primate animals. It's both technical and ethically very challenging to use these primates for these kind of studies. So if we can replace or reduce the numbers with these stem cell-based embryo models, I think that's a huge gain. So that, I'd like to thank you again with showing this picture again, and thank, of course, my fantastic lab and the unit where I work, and all the collaborators. And then I'll finish again on my personal sort of motivation for doing all this research. So after our marriage, we went back, and fortunately enough, we had embryos still sitting in the freezers. And we now have 2 beautiful daughters, and I spend a lot of the weekends watching them playing soccer. And this is my daughter scoring an amazing goal. So thank you, guys.
Thank you very much for an interesting talk and also personal talk. So we have space for one question.
Hi, Håkon Høgseth from University of Oslo. So thanks for a really great talk. I was curious, now you made these atlases of all the embryos. Have you spotted any feature that it's really hard to make at the moment or that is lacking? Like what is the type of features that currently we don't see or find in the embryo models?
I think I'm switching your question and sort of answering perhaps a little bit different. For our reference, what we're most challenged with is actually PGCs, because the references they're too few for us to really stably be able to project those. So we would need better, preferably in vivo references for the early germ cells. We're really lacking that. For the stem cell models, it's really a little bit different. I mean, but I think at this point we need to ensure that we at least have the major building blocks in our models, right? So we're not lacking all endoderm or all trophectoderm. But I'm not sure if there's like a systematic sort of tissue that we're lacking. And I think this is still very early, right? Then extending this into gastrulation and also, I mean, this model does not show ectodermal cells yet. And I think that's something that the field is perhaps a little bit surprised with, but learning that actually the ectoderm is forming later than we would expect. I would have expected to see that, a clear ectoderm signature already in the Teiser et al., the gastrula data set, but probably that matures a little bit later. I don't know if that was an answer to your question.
Yeah, that's good. Thank you.
Okay, excellent. Thank you again, Dr. Lanner. So now we come to some break, coffee and pastries in the front, and I'll be seeing you again at 11:20. Thank you. Okay, so hopefully everyone has got some coffee and pastries. Now we will turn to the second half of the mini symposium, and now we will go to the ethical issues. And first of all, we have Dr. Garðar Árnason. He's a professor of philosophy at the University of Akureyri, and Garðar's interest lies within ethics, in particular moral status, responsibility, and free will. And his title is on embryo models, potential humans, and moral status. So please come to the podium, Dr. Árnason.
Thank you very much. First of all, I want to say that I don't have a PowerPoint. There is no technical problems. It's supposed to be a PowerPoint. I want to thank the Nordic Committee on Bioethics for inviting me. It's a great pleasure to be here. And also want to thank the committee for organizing the workshop yesterday. It was very enjoyable and informative. And much of what I'm going to talk about here is informed by and sort of reflects perhaps, or is a reflection on discussions that we had yesterday. But having said that, the opinions that I express are mine unless I state otherwise. My title is Embryo Models, Potential Humans and Moral Status. I'm not going to say much about potential humans at all, but maybe more about about the moral status of embryos. What I want to look at are there are 4 questions I would like to address. I'm not sure I get to all of them, but I'll try. The first is, what is the moral status of the human embryo? Second, what time limit should we set on human embryo research? Third is, what is the moral status of human embryo models? And last question, when is a human embryo model equivalent to a human embryo, and does it matter? So 4 very big questions in a rather short time. But they are, of course, related. So if you have some idea of what the moral status of the human embryo is, you might also have an answer to the question what the moral status of embryo models are. And if there are problems, you might see also how that applies to that question. And also the moral status of the human embryo might affect how we think about the limits, at least the moral limits that we might want to set on human embryo research, and then also on research on human embryo models. So let me then start with the first very big question. What is the moral status of a human embryo? This is not a new question, of course. It's a long history in different contexts, of course, in abortion debates, lots of literature, and as you all know, there is no consensus, there's no definite answer. Also in the context of embryo research, at least since 1978, with the emergence of IVF, and at least the potential availability of excess embryos for research. So there's a long history of debates on the moral status of the human embryo, and I'm not an expert on the abortion debate or even the embryo research debate, but I want to say something about moral status that I hope it will rather clarify than confuse, and at least provoke some thoughts about the moral status of embryos and how we think about moral status of embryos. So what is moral status? What do we mean by moral status? What I'm going to understand it as follows. It's the extent to which someone or something has a claim to be included in the moral deliberations of a moral agent, ourselves, for their own sake. So moral status, the question of moral status is, why do we need to take the interest or the interest of something into our moral deliberations for that thing's own sake, or that person's own sake? So a pebble on a beach doesn't care if I throw it in the ocean or if I kick it. It has no interests one way or the other. I think it's safe to assume it's not going to feel anything. It's not going to mind. But if I kick an animal or throw a person into the sea, that person or animal will care, and they seem to have an interest in not being kicked or thrown into the ocean. At least they don't want to. So one possible account of moral status bases moral status on some moral relevant characteristics. There has to be something about that being that gives rise to that moral status. And in particular, it should be some sort of a capacity. So this is called the capacities approach to moral status. And I think it has been the most influential approach to moral status, and maybe also the most criticized, possibly. But the main rival to the capacities approach is the species approach. And according to that, humans have moral status or full moral status simply in virtue of being human. So one problem with the species approach is that it's not clear what gives the human species this elevated moral status above all other species. Maybe it's human dignity or the inherent value of human life, but it's very difficult to provide plausible accounts of those concepts, in particular as being somehow inherent in those in humans. So I'm very skeptical about this species approach, and I tend to go towards the capacity approach, and I have attempted to give an account of moral status in those terms in a paper a few years ago. But of course, that approach is not without problems. Two common problems with the capacity approach is first, what are the relevant characteristics? What sort of characteristics or capacities can give rise to moral status? And whatever those characteristics are, it seems that we'll get an approach that's very exclusionary. It excludes all those beings that don't have these characteristics. And those are often beings that we think should be treated as having perhaps full moral status, and I'll come back to this in a minute. So I'll try to see if there is a way to address these two problems, and then see how the capacity approach applies to human embryos. But I'm not a convinced proponent of the capacity approach, but I think it is one way of one influential way of thinking about moral status. And so I want to see how far we can take that. Now, common candidates for moral relevant characteristics that confer or give rise to moral status are often given as, for example, intelligence, rationality, language, sentience or consciousness or something like that. But it's not clear how these characteristics are morally relevant. And many of the proponents of capacity approaches don't give any account of how these characteristics are related to moral status. How do those capacities give rise to a moral status? But one way to bridge that gap is through morally relevant interests. So having a moral status is having a certain type of morally relevant interests that arise from specific capacities. And then we just need to see how specific capacities can give rise to these morally relevant interests. So as an example, an animal has an interest in not suffering pain. The relevant characteristic for this interest is sentience, that is, the ability to feel pain. And I'm using the term sentience here in a technical way, that it just means the ability to feel pain or pleasure. So animals, non-human animals, maybe not all of them at least, vertebrates probably, have the ability to feel pain, and they have an interest in not suffering pain. You might ask why, but I think it's fairly clear if there is anything that's morally bad, it's suffering pain. I think I don't need to give a further account of that. Now, humans have more complex interests, including in continued life, in freedom, in physical integrity, perhaps in happiness of some kind. And these interests arise from the combination of two characteristics: the capacity for consciousness and the capacity for metacognition. So now the story can get a bit complex, how the ability to be aware of ourselves as sort of unified beings in time, having memory of our past, being able to plan for the future. These capacities of consciousness and metacognition give us these abilities, which again give us an interest in seeing our plans through, in completing our projects, in remaining intact remaining our physical integrity, of being free, maybe seeing happiness and so on. How exactly in detail would be too long a story for now. But so this is sort of roughly how the story would go. So in that case, in this latter case for humans, we have something that's called full moral status or personhood. And personhood is here again a fairly technical term for those beings that have the sort of capacities that give rise to this complex of interests. So far, so good. No. At least it's a rudimentary capacity approach, but we, I think, already see the problems. The second problem I mentioned earlier about exclusion. What about humans who are not persons, humans who do not have these capacities? They seem then not to have interest in continued life or in physical integrity or freedom. But that seems to be completely wrong. And also the human embryo, the fetus, newborns, they don't seem to have these capacities. So do they then not have any moral status? This seems to be very problematic. And proponents of the capacity approach have gone different ways. Some just bite the bullets and say that those humans who are not persons don't have full moral status. So there have been papers suggesting that, for example, newborns or infanticide could be justified in certain cases, which also seems to, I think, to most people to be very wrong. Others try to save the approach, maybe by suggesting that the capacity is something, it's not just ability, but it's a capacity that we have, and now it's almost going into the species approach, in virtue of being human, we have a capacity for consciousness and metacognition. And even at the time before it's fully developed, at the time maybe in old age we have dementia or something that loses capacities, or even when you sleep or faint, we lose this capacity maybe briefly. We don't lose, well, we lose the ability, we don't lose the capacity that's still provides this moral status. But this it becomes somewhat technical and difficult, and we are now sort of on the problematic edges of the approach. But now also there are further problems. What about material objects that we seem to give some moral value? Like you may remember the Buddhas of Bamiyan, the big statues that in Afghanistan that were destroyed by the Taliban in 2001. That was a destruction of something that seems to be very valuable, but it doesn't seem to have any moral status, though. I mean, the statues didn't care, but it seems to be something that is morally wrong to destroy the statues. And wouldn't it be morally wrong to destroy the Mona Lisa or other works of important works of art? So moral status. As I said, the way I talked about moral status is that it's the extent to which someone or something has a claim to be included in the moral deliberations of a moral agent for their own sake. So the statues or work of art, maybe embryos, they don't have moral status for their own sake. They don't seem to have any characteristics that would give them any moral status for their own sake. But that doesn't mean that they don't have any moral worth, that we don't need to take them into account in our moral deliberations, not for their own sake, but for other reasons. So we might say that they still have moral value or moral worth. It might be based on some symbolic or relational value, might be based on some sort of moral intuition or moral emotion, social, cultural, or religious values. So rather than having inherent moral values, inherent moral status in this case, we can still talk about moral status, but it's the relational moral status. It's not something that's inherent in those things, but still something that matters morally, and often very much. So we don't describe the moral value in terms of some characteristics we find in those things, or these beings, but we ascribe value to them because of our relation to them, or what they mean, or because of how they are valued in society, or by religion, or for other reasons. So even if you have a capacity approach to moral status in some sense, it doesn't have to exclude other sorts of things mattering morally. So I think, at least on this account, the human embryo does not have any inherent moral status, but a relational moral status. So the moral status of the embryo may very well be gradual from fertilization to the fetal stage to full term in this sense. But how do we know then what moral status it has? And if this account is plausible, the only way to find out, it doesn't really make, we cannot find out only by investigating the embryo. What we would have to find out is what sort of relation we have to embryos. We have to ask people. We have to consult ethicists, lawyers, scientists, theologians, religious leaders, those who have some stake in the research or in the matter, in embryo research with patient groups, and of course general publics. And this is not just a matter of balancing moral opinions and values that are all somewhat equally valid. These moral values and moral opinions, they can be poorly informed or poorly thought out. They may be better or worse justified. They're always open to debate and discussion. So that's, I think, one way of approaching the question of the moral status of the embryo. I think it's very difficult to find any moral relevant characteristics in the embryo that would give rise to a moral status for the sake of the embryo itself. Well, of course, I may be wrong. But in any case, this seems to have, at least the question of inherent characteristics, might also have some import for the question of the time limits that we should set on human embryo research. And this goes back to the embryo research I was talking about, arising from the emergence of IVF, the excess embryos that became available. There was first suggestions in the United States already the year after 1979 that there should be a 14-day limit on embryo research. This was taken up in the Bona report in the UK somewhat later. And this has become, as you probably all know, sort of almost the consensus that embryo research should be allowed at most until 14 days of development from fertilization. Until 2 years ago, when the International Society for Stem Cell Research revised its guidelines and suggested that perhaps it should allow, or it permits, embryo research past the 14 days on case-by-case basis. They didn't suggest any other time limits. And the I think there are some good reasons why, but in a paper by Hank Greeley that was published last year, called 14-day rule, a modest proposal, Hank Greeley suggests that that it was a serious mistake to not propose some limit to embryo research. And he thinks that mistake is very understandable because it's very difficult to set a limit. And he tries to do so himself. And he first looks at the different stages of development to see if there are any particular developmental stages or points on the stages that where you can draw a line. And he has 10 suggestions where you could draw a line, a limit to embryo research. I'm going to go through these suggestions. I think they are interesting for various reasons. The first is the appearance of specialized tissue that will become the central nervous system about weeks 3 and 4 post-fertilization. So he is looking at things only after the 14 days. He thinks you couldn't set a limit at fertilization. Someone might want to do that, but we are talking about expanding the limit after the 14 days. So we look at any reasonable points after the 14 days. So one is the appearance of specialized tissue that will become the central nervous system about weeks 3 and 4 post-fertilization. Just a note on these times. He refers to, and I think the discussion has mostly been using this time frame of post-fertilization. This would be, in weeks, this would be, if you're thinking in terms of pregnancy, the stage of pregnancy would be 2 weeks additional to these times. So the time frames I'll go through are all post-fertilization. Anyway, appearance of specialized tissue that will become the central nervous system. Hank really doesn't think this is really plausible for any emergence of any specialized tissue that would become something else. It's hard to see how that could matter morally to anyone, really. Second option is detection of electrical impulses that will later signal a heartbeat. It's about week 3. And that's an interesting one. In the abortion debates, I really don't want to go into the abortion debates, but they cannot be avoided sometimes. In the US, some people really put a lot of weight on the heartbeat in the embryo, the fetus. And of course, what was sometimes referred to as a heartbeat is just a pulse of a cell. There is no heart yet, so it is the pulse of a cell. But still, that seems to raise some moral feelings in many people who observe this pulsing in the embryo. But still, it's hard to see how this can have any really be of any moral interest and make any moral difference. First apparent blood flow through what will become the heart end of week 4. Same thing as before. Why would this first flow, this first sense of blood flow make any difference morally? First, differentiate neurons with synaptic connections late in week 5. So the neural system is developing, but why would that particular point be of moral interest either? Like the other things, where some parts, some cells are just becoming something else. Doesn't seem to be really that morally interesting. Fifth option, first neurons in the developing brain, the start of week 7. We might start to be concerned about sentience, like being able to feel pain or even consciousness. But of course, that's not going to take place by when the first neurons emerge in the developing brain. So that doesn't seem to be really of moral interest either. So those are the first 5 points. 6th point, change of name, nomenclature, from embryo to fetus, end of week 8. Hank really suggests that there isn't much more happening there than just a change of name. At that point, perhaps the organs are all in place, but that doesn't mean that they are functioning. They're not, of course, not fully developed, and most obviously the lungs are not functioning until the child is born. this doesn't seem to be really of any moral interest either. Beginning of ability of the fetus to respond to sound, week 16 or later, there are some conflicting research on when this happens. Again, this might suggest that there is some kind of consciousness or awareness, but apart from that, still not really that morally interesting. Viability about week 21. This is perhaps something that many people think, at least here, and Hank really thinks, at least at this point, we shouldn't be doing anything to human embryos. They could practically live, they can live outside the uterus, that's what viability means. They can, of course, not live by themselves, even newborns cannot live by themselves. But viability means then that they can live outside the uterus. This time frame is, of course, changing, and with developing technologies, this might become earlier and earlier. At some point, it might meet artificial uterus coming from the other end. But in any case, viability seems to be something that might matter to us. Ninth option, beginning of the ability of the fetus to experience painful stimuli about week 27, becoming sentient. Here, at least, we have inherent moral status. It's something that, I mean, the embryo, if it can feel pain, seems to have an interest in not suffering pain. And it would be wrong to inflict pain on it. Full term, about 38 weeks. Again, here, I think, apart from the most hardcore capacity approach proponents, we think definitely at this point, it would be wrong to be doing invasive or risky research, except in very specific exceptions. So the main point here, for Hank really at least, is that the viability seems to be the only point where you can really say after this there shouldn't be anything taking place. That's a very sharp, something that should matter morally. But even then, he cannot really give any good reasons for it, except for his feeling about it. Is this, yes, moral intuition or feeling about it, and he is very. I think he's in the paper. It seems to disturb him a bit that he cannot really give any good account of why viability makes that much difference to him, but it does. But he has a solution to all this. He asks us to forget about all these developmental markers. They don't really help us very much. We should instead ask, what is the most pressing scientific need for embryo research? And many of you, maybe all of you, are familiar with this. That is the black box or the dark zone in embryo research between 14 days and until miscarried or aborted embryos become available for research about 28, 35 days after fertilization. So in this window there is very little to do research on. And so there's a gap in our knowledge of human embryonic development between those dates of development. And then nothing seems to be happening during this time that's really morally relevant. So we should look at the pressing scientific need in this case and expand and set the limit then at 28 days for that purpose. There are, of course, concerns with this sort of suggestion, I think. One is a slippery slope. If we start expanding it to 28, scientists will come again and say, well, we need to expand it more and then more and more. At some point, they'll be doing some horrible research that shouldn't be allowed. And again, we could take the other, the opposite concern. I think that this is too narrow. Why should we stop at 28 days post-fertilization? There isn't really anything there either that should give us concern. So why not do without a limit or put it much later if there is scientific benefit for it? But still, there is an assumption, there is a moral cost to embryo research that grows gradually with the development of the embryo. There's some growing moral concern through its time. And the question is how to estimate this cost. How can you figure out what is the cost of what is this growing value, moral value of the embryo and then the fetus? I've suggested that we need a discussion of the values and moral opinions that we have. There is no easy way to do this. And then there is a further concern how, even if you can get some idea of the moral concern, how do we balance that against scientific need, scientific progress or benefits? How do we evaluate that, and how can we compare that to the moral concerns to make a decision? So I'm just leaving these as problems, as concerns that we need to think about. And move to my third question, which is probably, it will be the last thing that I can, the last question I can address. I've suggested that human embryos and then also human embryo models, they don't have any inherent moral status. There is nothing really, they don't have any characteristics that make them matter morally for their own sake. There is some relational or symbolic or intuitive value. There is maybe value of human life that we ascribe to them, but it's not something that we find to them. So if they become equivalent to natural human embryos, it seems that they should have the same moral status. But here we could even ask, is that really the case? Some people might think that since they are not natural embryos, they are more artificial, synthetic, or as the terminology goes now, they are derived from stem cells. They might have less value than actually natural, real embryos. Or the opposite, if because they are created, they might have more value. The researchers might be responsible for their creations, as Anna Smyter suggested in the workshop yesterday. So that might give the embryo models actually more moral significance or a different moral significance than the natural human embryos. There are also questions about the different kinds of embryo models that we have discussed earlier, in the talks earlier this morning, which is something that we could discuss, but I'm going to stop here and maybe take a question if there's time. So thank you very much.
Thank you very much, Mr. Árnason. Is there a quick question? A quick answer? Okay. Yes, Lanner?
So if I put you on the spot, what limit would you say? Impossible question.
What limits? I have a lot of sympathy with Hank, really, that I think I would go with him and set a limit at viability, but not really having any good moral reason. Even going beyond that might be justified in very, very specific circumstances. But I think in terms of the approach I was suggesting, there might be a question of when there is a capacity for these characteristics I talked about, like consciousness, even emerging in the fetus without it being conscious, but there is a capacity there that's emerging. This might give us a reason to ascribe a full moral status to it. But I don't. I think. Sorry. I think if there is a strong scientific reason for it, that might be. But again, I don't think it's not going to be my decision. That's maybe the point that I wanted to make here. That we need to get a public discussion, social discussion about this issue to inform where to draw the line. And I should not be making the decision. Thank you.
Thank you so much, Mr. Árnason. So then we will turn to our last speaker, but not the least. Her name is Dr. Johanna Ahola-Launonen. She's a postdoctoral fellow at the Aalto University, Helsinki, in Finland. She is specifically interested in social and moral philosophy. And she will be talking about beliefs and interests concerning embryo models. So please, Johanna.
I will start by making myself visible. Hi. Uh-oh. All right. Then I'll... Oh, the table clicked the mouse. Let me see if I can handle this. In the meanwhile, I'll select what Garðar just... Garðar started by thanking you all at the Norforsk and other parties and saying nice things about this event. So I'll select and copy and paste into the beginning of my talk here. Okay, so for now on, we've been talking about the embryo models, and I want to take a step back and discuss the ethics of the beliefs and interests that we are giving to the embryo models. So the expectations that are raised towards the embryo models. And it's not just us here, but it's how the issue is discussed to the public, to the media, and what kind of expectations all this discussion kind of raises nationally, globally, in media, in public, and so on. So on the very technical subtitle here, it could be replaced bio more like a explicit subtitle of how to manage expectations in a responsible manner that does not create over-enthusiasm and costs of hype. So let me start by kind of giving, my intention is to give some tools for thinking the issue. And let me start by just like framing two different kinds of units of analysis when we discuss the implications, when we analyze the ethical and legal and social implications of what. So kind of the traditional way to think about it is that we think about the ethical implications of the artifact, the technological artifact. I know this is a CRISPR picture, so embryo models were not, I could say they were not available, but the truth is that I just didn't remember to change the picture by the time I sent it to Bettina. So please, you can use your imagination and change it to an embryo model. But actually it doesn't matter, because the point also here is kind of a look back and kind of learn from hype lessons that have been taken place from other emerging biotechnological stuff and the sorts of expectations that has been risen by those, and what we can learn about those hype cycles that has preceded this emerging hype cycle with embryo models now. Anyway, this is the usual way to look at the technique, technological in a wide sense, as meaning as a technique that humans or persons use to build their like benefits or human flourishing or whatever. But the other analytical unit of analysis is to see that what brings about this artifact. And that's called the sociotechnical system, including all sorts of political and cultural and economic values and decisions and all that. So in other words, the beliefs and assumptions and valuations, the historical, the political, the cultural and the social context, all the dynamics of power and interest, all the economic and scientific decisions of science, politics and framing the research agenda that kind of preceded the development of the technology or the technique, and that maintain the wide context in which the technique is seen as an important answer to something. So here you have kind of two different sorts of units of analysis when we think about implications of stuff. So the questions with the sociotechnical system would be that what are the implications of these beliefs and assumptions and valuations and relations of power that serve, what are the implications of all this that serve particular interests and decisions and prioritizations? While when we think of the technological, of the artifact, the question is that kind of usually, especially when we think about emerging biotechnological, well, I'll call it stuff. The question often is that we ask that what follows when this technology exists and will be in use. So we kind of look into the future and speculate and forecast the moment in which the technique or the technology will exist and be in use. And there are several problems with this type of thinking, because by doing this kind of future prediction, we kind of reinforce the belief that favors developing the technology. If we forecast the future and take the technology as a given, so we kind of we take it as a given that this certain type of artifact or technology will exist there in the future. So that type of thinking reinforces all the sociotechnical system stuff that kind of maintain the context that favors bringing up this technology. And it also kind of advances a very linear and optimistic view about how science progresses. And we all know that science, the progress of science, it's not linear. It's not straightforward. It will not, the scientific progress will not go as we hope it to go at the moment. It will take detours. It will fail. It will find another path to something else. It will take time. So focusing only to the future existence of the artifact is problematic in many ways. So the questions that should be asked include, for example, that what do we assume that when we assume that an artifact A is a good answer to problem B? A very good example is that when we talk about embryo models, we do have like very specific expectations listed. For example, we expect to have more knowledge about early miscarriages. But also there, in the side, there's the rhetoric of expecting the improvement of human health, or the improvement of human well-being. So if we assume that embryo models will be a good answer we want to improve human well-being. So the problem is that humans are ill-being at the moment. And if we take the artifact A to be a very good answer to improving human well-being, so what values are behind these logics, and who benefits most of these valuations, and what other solutions to improving human well-being would exist? Are the resources well spent to artifact A to improve human well-being? The question is to be able to critically ask that which science is the one who has the best answers to improving human well-being. Is it the social politics? Is it the subhumanities? And so on. So when we speak of used intellectual and economic resources, what's the basket in which we want to invest? Of course, I'm not trying to be critical towards embryo model research, of course, but I'm just lessons learned from, for example, from the Human Genome Project have taught us that the expectations that were brought up with like the Human Genome Project and how like all the diseases will be cured very soon, and we can make humans more intellectual, and we can enhance some moral genes, and all these kinds of very speculative and very unscientific expectations have led to this sort of very unscientific genetic determinism also in social politics. So those expectations that were brought up with the Human Genome Project have had political influences of how we understand the construction of health and well-being, or have kind of contributed to that. And most importantly, it's important to understand hype cycle phases. I'm going to talk that next. That in which phase of the hype cycle are we with this specific emerging biotechnology? So in contrast to these questions, focusing on the artifact would be to ask that what implications does this technology or technique have to these and these parties? Anyway, looking at technology hype cycles, this hype cycle construction is from a kind of a commercial and commercial consultant company looking at commercial technology development, but this is very applicable to scientific process, at least to some extent. So first there's the innovation trigger in which we are kind of at the moment, for example, with embryo models. There's some new knowledge and expectations that we are going to be able to do something big very soon. And then with all new stuff, the hype starts to build and build and build and build and build. And then it reaches the peak of inflated expectations, where the hype is like, as it's like the biggest phase. And then starts the true of disillusionment, when we start to know that, for example, in the Human Genome Project, after the whole genome was sequenced, we learned that, well, actually, we only now have a lot of data. We didn't find the cures for all the diseases very soon. There wasn't any gene per complex traits and so on. So the reality starts to kind of come in. And then the rest of the slope of enlightenment, like we start to understand the value of the knowledge that was received. And then in the marketing phase, it's called the plateau of productivity, where blah, blah, blah. But I'm not sure that's purely applicable to like scientific process. But anyway, the most important thing here is to understand how the costs of hype build with these kind of hype cycles. And my point here is to kind of contribute to that we could escape as many costs of hype with this emerging stuff right now. The costs of hype include, for example, promissory marketing that offer a vision of the future that may never be realized. False expectations of therapeutic promises that will be just around the corner. And also decreased trust to science is also a cost of hype. When patients and the public learn that, hey, nothing came up, or at least the stuff that I expected didn't come. These weren't made. So it decreases trust to science. The scientists said that human well-being will be improved, but I don't see any improved well-being here, so it decreases trust. And then simplistic problem setting is also a cost of hype. If we contribute to the belief that only by biotechnological means human well-being could be improved. And then there's the question of inefficient or unfair or just bad use of resources. So if we think of the resources that are available to scientific research, all the sciences and social sciences and humanities taken at large, so we must be able to ask that what's the best way to use all these resources, and what kind of world do we want to build up? Which are the most pressing societal needs? What answers are most pressing? What tools from which sciences are the most pressing ones? So these are costs of hype that emerge when the scientists might promise something a bit too much, and then the media or if they are like the ones in the lab or at the institutions or universities who kind of publish some popularized versions of the journal articles, and they build the hype in there. Then the science journalists read that one and they continue to build the hype even more to public media, and then the public reads it and uses their imagination and starts to like fill in all these kinds of expectations. And that's the kind of the very integrated way how we reach the peak of inflated expectations. So the question is that how could we manage these expectations with embryo models at the moment? So we must kind of ask ourselves that what presumptions are made right now, and what promises are made right now at this moment where we are kind of in the innovation trigger phase with embryo models. And then what actually is expected. And yesterday I spoke a lot about the need to have very precise promises and very precise information from the scientific community that what we are doing and what do we expect. And I call for specific expectations. So instead of promising this very general improving health, improving the human condition, making the world a better place, so I call for specific promises of increasing knowledge about infertility, increasing knowledge about the black box in the embryo development, increasing knowledge about how embryos respond to certain medications. Those are specific promises, or some specific promises about the potential stem cell work that can be done with the embryo models. And then again, looking at the Human Genome Project and what kind of a peak of inflated expectation that caused, is that the problem is that it's still going on. When it started, since it started, the knowledge of how human genetics actually work, how the whole theory of the black and white division between nature and nurture has been kind of collapsed, and how the genetics is currently understood as a very, very dynamic and continuously interacting system between the genes and the environment. But still, that knowledge kind of hasn't reached much of the public still believes in gene pair, like believing that humans would have like one intelligence gene that could be enhanced by doing stuff, or that there would be one gene for alcoholism or criminality or all sorts of diseases. Well, of course, there are the diseases that only have one or two, and those could be cured or like manipulated. But most of the complex traits are like way out of that discussion, and still the peak of inflated expectations that were brought up with the Human Genome Project kind of still persists in public health, social policy discourses. So it's a serious matter of taking responsibility of how to communicate expectations. So here are my kind of suggestions for the ethics and management of expectations. Just to kind of restate myself. Focusing on giving relevant and precise information to media and to public and others, including other scientists. Because as we learned yesterday in yesterday's program, even us scientists from different fields of science, understanding science, including social science and humanities, even we had problems of like defining and understanding like what are we really talking about, like when we speak about 28-day embryos or whatever. So it's a serious matter of being able to communicate the relevant information. Then responsible horizon scanning and communication strategies. So how the scientists kind of scan the horizon, like scan what we can reach with embryo models. Like I call this the funding application language. When you write the piece about the societal impact and you make and you promise to save the world, but that's not okay as a communication strategy to anyone, because that is what creates the costs of hype. And then I kind of call us on keeping away from extensive speculative ethics and science fiction ethics, and both with potential benefits and harms. So when we speak about embryo models, if we don't have a precise information that what is it that science would like to do, the problem is that it's an open-ended question. And if the question is open-ended, we will use our imagination to imagine 7-month fetuses in tanks from Matrix and all that stuff. People can imagine potential harms and benefits with our brains filled with science fiction imaginaries. So, yeah. And then keeping the unit of analysis both in the artifacts and the sociotechnical systems. So it's kind of restating this. So not only thinking that what implications would the embryo model have to the embryo model, but also what kind of implications my expectations could have to the society, to humankind. So, yes, this is my final slide, and I think we have room for questions. Thank you very much.
Excellent, Johanna. Thank you for a clear talk. Are there any questions in the audience?
Many thanks, Johanna. I'm not sure if I fully agree with you that I see a lot of hype in this area. Mm-hmm. So I haven't really seen any promises of anyone saying they'll save the world. If we look at Jacob's and Fredrik's presentations earlier today, I think they were quite precise in their promises. Also, I feel like the things they brought up, for instance, drug toxicity screening, that is very much in the works. In the US Congress has already changed their laws to allow for the use of models instead of animals in drug trials. And actual work is ongoing in this field. So it's not science fiction in any sense of the word. So where do you see the hype? Like, who's hyping this? Where exactly is the hype? That's my question to you.
Yeah.
Thank you.
Thank you. That's a superb question for me to clarify my point of view. I'm doing preventive medicine here. So I'm not stating that there would be, because we are in the beginning of the innovation trigger. And my intention here is to prevent that this emerging thing would reach like a huge peak of inflated expectations. I'm trying to prevent the costs of hype here. And therefore, I'm calling for all this stuff in order not us or the kind of, because it's so easy to just like, you need to focus a lot for not saying the promises about improving the world and improving human well-being, because those are like the usual and jargonist ways of kind of defending the research agenda. So it requires a lot of attention for not building the hype. And this is the moment, the exact moment where we can have an influence of how we start to speak about the promises of this new practice. But you are absolutely right. I am not accusing. I have heard improving human health, and I have heard improving human well-being during these 2 days, but not as a very over-promissory. But this is preventive practice. Thank you.
Yes, Vilhjálmur, please wait for the microphone. So Vilhjálmur is here in the front.
Yeah, thank you very much, Johanna, for an interesting talk. You talked about hype, but couldn't the expectations and sort of discourse also be, downplaying and with fears and wrong-headed information, about crazy scientists doing something very irresponsible, something like that? I mean, what about that?
Yes, thank you. That's also a very important question, but that's also a cost of hype, because if we start fearing that stuff, the precedents. We need to have those fears. We need to have an imaginary of the tanks, of the possibility that there would be some really alarming stuff going on, and that's related to kind of the building hype. So hype is always like there are the people who will love it, the science fiction, and then there are the people who will hate it and fear it. And both of those sides are costs of hype. Thank you. Yeah.
Any more questions to Johanna? Okay. Thank you very much again.
Thank you.
So I want to say how grateful I am for the quality of the presentations that the four speakers have given us today, and I would like to give them a big applause. So now we will turn to the last part of this mini symposium, which is the panel discussions. And the moderator of the panel discussions is Dr. Sigur Kristinsson. He is a professor of philosophy at the University of Akureyri and a former member of the Nordic Bioethics Committee. So I welcome you. Please come.
Right. Hello, everyone. So there will be no panel discussions without panelists. So I would like to please invite the 4 speakers to take a seat in the panel. Right. And just to reiterate what Guðrún said, I want to also express my admiration for the 4 talks and for the discussions yesterday. I have learned a lot from this. This has been much information and also different approaches. Sort of people are coming from different directions and backgrounds to try to discuss a matter of common interest. We did today organize the talks so that the science came first, and then the sort of social and ethical issues afterwards. And in between we had a break, and that's maybe symbolic of just how these things often work. There is a gap sometimes between these 2 worlds. There's a different terminology. You need a lot of initiation and training to just to know the terminology and the basic facts, for example, about the human cells and fetal development and so on and so forth. And the same with the ethics. And so there's so much theory and learning behind all this. So it's really no wonder that the public discussion sometimes is not ideal, but whether we like it or not, there has to be a public discussion because there has to be a regulation, and the law will be in one way or another. So based on that, I want to first ask the panel, give the panel an opportunity to say something and then open up for questions. And sort of my triggering question to the panel is if they want to take turns responding to, well, 2 things. One, if there is something in particular that they would want to say to the other panelists as a response to what they've heard this morning. And then, secondly, well, what are their hopes for a discussion and a way forward in terms of, and then I'm thinking in terms of the legislation, the 14-day rule, and also ways forward in the science itself. So these are really, really broad triggering questions, but hopefully something that you will catch up on. So maybe, Dr. Hanna, if you want to begin.
Yes, I'll share something, perhaps it was less on the ethics, but something I already shared with Fred, but I think I want to share with the audience is, of course, when we try to talk about models and what's a model and what is why, there is obviously scientific merit, but it's very important because when it comes to regulation, it's which ones do really have the potential to do the things we want to prevent or want to look at. And for example, when we see very important single-cell RNA-seq data and looking at things, to me that has to come after the structure. Because in this case, the technology doesn't tell you. It tells you which cell types, tells you maybe how they look, but it doesn't tell you whether this is actually a structure or it was just, you can just take the same mess and it will give you the similar result. So, and I think one of the things when it gets to define what is also, when you talk about an embryo model, is the S cells are an embryo model. Embryonic bodies, there are systems or gastroloids are also embryo models. But clearly I would say mostly that they don't have the potential to make an embryo. Versus I think, and it could be different criteria, but we need to think about it like what is the role of the structure, what is the role of the signature, what is the role of the cells. I don't have an answer for, but I'm just raising, we're all in this dilemma in a way. And it's not just a bit kind of just being nerdy about it, but I think it is important because it's I think will dictate regulation, because you don't want to impose limitation on things just because they fall under this very general term, but actually biologically it's just they cannot do this.
Okay.
And if I just
Thank you
quickly
very
can
much.
respond,
Yes. Yeah.
I mean, I completely agree that sort of you can't sort of just evaluate based on gene expression profile, but it's also apparent that you can't evaluate just based on morphology.
For sure.
So, I mean, we need both of them.
So, yeah.
And then, so it's like morphology, gene expression, like profile, and then ultimately function also.
Yeah,
right?
yeah.
So all those, and I don't think you meant this as a critique against
No,
me,
no,
but
I'm
I
sure
just want to sort of also add, right, that morphology in itself is not
No,
enough.
not at all.
And as developmental biologists, we've sort of focused a lot on finding specific markers that identify different cell types, and I think we need to go away from that and more look at like fingerprints or signatures.
And it could be spatial
then link it with-
transcriptomics also then might give you that input, but it's definitely multi-layered.
Okay, thank you. But so, Fredrik, did you want to then have a more general response or question or response to the other talks, or thoughts about what your hopes are for both in social terms and scientific?
I think both the discussion about Hank Geely's sort of trying to find a time point where it's sort of not ethically okay to continue. I think I mean that is to some extent like the core of what we're talking about here, right? And yeah, I don't really know if I have much more, but I mean I think it's we're sort of discussing whether we should extend the 14-day rule, and then we have the view that actually there may not be sort of an ethical dilemma until week 21. I mean, the sort of the gap between these views are just enormous, and it links back to I think a comment that you made. I can't remember if it was this morning or at another point, that we're trying to sort of rationalize the limits by saying, okay, we have this black box from implantation until we have aborted material that we could study. And I think I've been one of those people that sort of proposed that perhaps the 28-day is sort of a good limit from that perspective. But then on the other hand, I also agree with you, if we really want to sort of do something more than just characterize how the tissues look like, what the genes are expressed, how the genes are expressed, and sort of functionally understand, it's not going to be enough with those terminated sort of pregnancies. And then the question is, what is it like? What's the core that really prevents us from not going beyond even 28 days? So I also don't have a solution, but I think also yesterday Nicholas brought this up also, that taking progressive small steps might be one sort of pragmatic solution to this, and we can't completely predict what's going to be in the future and where we are in 5 years or 10 years time, but I think it's quite clear from my perspective that we need to sort of update, or at least consider to update the current rules, and then stepwise progression might be one strategy. But I see your point also, and then of course we should consider that this is not what we're discussing in Scandinavia and the Nordics is not the same everywhere in the world, right? So whatever we decide, it's another reality for you or in the US and so on. And I can't resist a little bit. I also got when you brought up the sort of discussion of hype, and I think this is really a shared responsibility, as like from the biological scientific sort of perspective. I think we're always trying to downplay, and we're really critical about like what this could potentially generate and so on. But then as soon as you try to discuss that with press and it's always like tweaked a little bit extra, and then sort of and in these discussions also, right? We're always going to the most extreme, and that was the same with genome editing discussion. You're always going to the most extreme. That like you're gonna design people that can fly with wings and so on, instead of thinking about like trying to treat a certain disease. So so it's a shared responsibility, and to some extent we might be overselling our sort of promises, but my feeling is that we're really trying to like downplay it and sort of say that there's like the models are imperfect and there's a long way to go and so on. And I don't think you meant to blame only the scientists, right?
Yes.
But it's
Right. Thank you. So maybe we'll just move to Johanna.
Yes.
Yeah.
I actually did mean to blame also the ethicists very much, because there's, well, with the human genome and gene editing, like there was like this blow up of philosophical applied ethics literature on like playing God and like and this and like enhancing this and that, like a whole scene bursted up, like visioning these futures where we can morally enhance and biologically enhance and all this what I would call science fiction ethics, that then people actually believed that could be scientifically done. So I'm very much like calling to the ethics people also to kind of take responsibility of because in philosophy the line is very thin between like conducting philosophy with thought experiments, which is a valid practice, and then like imagining like or like taking the thought experiments for real, as if we could build wings and stuff. So it's a thin line, and I think that the ethics community should have more responsibility of like of which kinds of visions they kind of speculate with. But so that's really a shared responsibility.
I would add also journalists to that.
Yes, exactly. Yes, journalists are also like sometimes in the worst end, like of
clickbait.
Exactly. So I'm kind of thinking with this because, also this including this embryo model topic, it's the questions, the fears and so on can be like really, really bad. So I'm kind of thinking whether like I'm just visioning these kind of minutes to journalists, or like a handbook for communicating about embryo models, updated in every 6 months or something like that, with like please be cautious when you communicate, like please do understand these and these facts, and like with every press release or whenever communicating with a journalist or with an ethicist, you would like approach with that kind of sheet or like information sheet, like please take your responsibility of something of a practice like this. I think should exist, because otherwise it's really arbitrary of who's the one who's writing the article. Mm-hmm. So there should, in general, there should be this kind of a practice of responsible communication. But then I guess some people might come and there are people I know who say that it's like censorship. Like, I'm a philosopher, you cannot censor, I can write whatever I want. And or journalists, like why should I censor my visions? But there should be a kind of a elaboration of that responsibility.
Good, thank you. Garðar, what are your responses and your thoughts about where to go from here?
Well, just to pick up on the first thing that you said, Sigur, I think it's really great that we have here science and ethics, even if we have this gap of a break and disciplinary gap between them. And as for the 14-day rule, I am personally very permissive. It means that there's enormous gap between viability and maybe 28 days of development. And of course, I should not be deciding this, but what we need is a discussion in society about these things, and it's important that scientists also take part in that, and therefore science communication and the participation of scientists is important. And I think Johanna has made a really great job of also showing us the difficulties and concerns that arise there. And as for my hopes, I hope there will be a continued discussion of these things, both in fora like here and more widely in society, and then well-informed and well-informed and rational debate about these things.
Right. Thank you very much. So I'm sure that there are people in the audience who would like to participate. Yes, we already, let's see, who has a mic? Yeah.
Thank you very much. So regardless of which day we end up with, do you guys believe that work on complete embryo models should be subject to the same day limitations as research on real embryos or not, and why?
Is this a question to everyone or to...
It's a question to everyone, but first and foremost to Jacob and Fredrik.
Yeah, I think so. As I said, it's a complex question because you're immediately jumping to a scenario when we know that they are perfect or 95% perfect in that aspect. And then you have another layer of complexity, which is, as I mentioned a little bit, implantation. So you can't, a lot of these structures, let's say day 20 or 30, even if they are they're excellent, you can't put them back into uterus. It would just never work. So you have also that middle layer. So you might have an earlier structure, which is a blastoid. That one, if you put it into uterus, is different. So that you already have this difference already happening there on the level of putting inside the uterus, if for some reason, although it's illegal anywhere, but let's assume for some reason you have to, on that aspect. And the number, and then if I jump to say what is the layer, if it's the number of days, and then I mentioned, well, you also are going to have the option of soon limiting things that are not viable, removing no neural tissue or removing no heart. So you have these complexities, which you have. So I think I can imagine criteria, well, but if that thing doesn't have a neural tissue or heart, it's not going to be treated similarly like the other one. That is one thing. You might have a complexity, if that is, well, you say if that is protocol that is life-saving to do from the donor, then you might say yes, but for basic research, no, although you might don't. So you have, I think I can imagine the criteria, not only, even think it's a multi-layered thing, and then you have, as I said, the different countries and different cultures have different approaches to what limits they want and what they consider viability. In Islam and Judaism, life begins after birth. So how do you deal with it? And I'm not saying that they really so it's complex, but as got mentioned here, the good thing is the progress is, I don't think that the progress is that fast, actually. You're going to see it's very slow. And so as we go through, I think these things are going to develop. What Nicola says, he suggests these small steps. I just think the small steps are just inevitable. It's going to be by small steps anyways. So, and then we kind of, I don't know, grow into it or meet into it and see what happens, but in a responsible way, not, we just leave it. I think, and maybe there, and again, I say discussion, different bodies. The UKs now have a body of governance of embryo models, and I know the European Union, in terms of the ERCs, are also starting to think about this. And so all of this together, I think it will slowly but surely kind of converge in each environment. Again, I'm sure that I don't think it's possible to have universal. I'm not even sure it's wanted.
Right. Fredrik, did you have something to add to that? Or?
No, just that I'm sort of. I would not really. I mean, I think at the point now we can't really say that these stem cell models are embryos because they are just so distinct. I mean, the suggested sort of steps that Nicholas sort of talked about, like at what point is it a tipping point or a turning point where they could become an embryo? I don't know how we could write that into legislation today. So I think we need to keep these sort of models in a separate category, and whether it should be sort of legal limits or if it should be sort of restrictions for like oversight in some way. But I think we need to keep them right now at least separate.
Mm-hmm. Mm-hmm. Good, thank you. Erna, right?
Yes, I was just, maybe because Garðar was talking about the, yeah, thank you.
Maybe, I'm sorry, maybe I'll ask you because I forgot before just to say your name.
Yeah, I'm Erna Magnúsdóttir. I'm an associate professor here at the Department of Medicine.
Right.
So, because you were talking about that maybe the limit should be at 21 days, like at the point of viability. I wanted to kind of play the devil's advocate a little bit, because you might have embryos that are never gonna be viable outside of the uterus, because and we don't actually even know if the stem cells that we're using now, and we talked a little bit about it yesterday, that we're limited. Most people are just using the same stem cell lines. We don't actually know if they make viable embryos, if they're genetically competent. And you could picture a scenario where we would actually have genetically incompetent stem cell lines because we knew they would never become viable outside of a uterus if they were implanted. So maybe that's actually not even a good limit. And this is not just kind of fiction, because already like with the CRISPR scenario, the first embryos that were edited were actually taken because this was a Chinese experiment that they were infected by 2 sperm, and the first the justification of that experiment was that these were never going to be viable embryos. So I'm thinking, if this is the kind of cutoff, they're viable, then I'm thinking, then can we get around that limit and going further just by taking something that's never going to be completely viable?
Right, so we could have engineer Fide so that it can grow even to full term without ever being viable, and then we could do whatever research we want with them. I might even not have a problem with that, but in the end, it has to be acceptable to society. I don't think that will ever be, very unlikely at least. And I think that's the important thing, that in the end it has to be acceptable to society, what's being done.
Yeah. Maybe says, please raise your hands just as I will scan the audience, but maybe I'll just pick up on that and ask you, so because we're talking about facts and norms and sensibilities and emotions and emotional sometimes reactions to images and ideas, actual or imagined, and so on. So it all becomes a big mess. It can be that in the public discussion. But then this somehow in a democratic society is supposed to end up in some kind of regulatory regime, right? That has legitimacy because it's based on sort of norms and some sorts of legitimate processes. So one way one question that arises is just, where do these norms come from? I mean, you might have norms that say that, well, you know, life begins at birth, or norms that say that life begins at conception, or anything in between. So what, I mean, do we take a poll, or is there are there particular sections based on expertise and roles in society that have a privileged, sort of authoritative voice when it comes to what the role should be. Religious leaders, politicians, scientists, ethicists, whatever that definition is, what's an ethicist? So that's my general question, but I'm thinking mostly about scientists. I mean, because it's understandable that scientists want to continue to move the boundaries of knowledge and maybe just try to figure these things out because they're unknown. And it's just important, and you're driven by curiosity maybe to some extent. So, but then there are sometimes the norms come from the outside. But your curiosity cannot go too far because we don't want to allow you to perform experiments on things that we consider sacred. But what do you have thoughts about what the role of the norms that arise from the scientific practice itself should have? Or should they maybe be in an authoritative position above anything else? I'm not sure this makes sense, but if you want to respond, anyone, to these thoughts, where do the norms come from, and what's the role of the scientists in articulating them and putting them forward?
Clearly, the scientists should not be the authority, of course. I mean, that's not the way to go. Mm-hmm. But of course, we have a role to sort of describe our view of the world and how we see this mm-hmm. type of sort of potential and risks. And then, but I think I sort of raised that yesterday also, that at least in Sweden, I think it would be very useful to try and do some sort of poll to get a feeling for like what is the current sort of feeling in acceptance in our society now, and would it change with more information or not?
Right.
Yeah. What a big question. I think that like my first idea is that we should also remain from like drawing these stereotypical like images of the scientist who'd always want to go just further and further. Like somehow I doubt that the mass of scientists would want to have like these 6-month-old fetuses in tanks in labs just for the sake of knowledge. So like scientists are also humans, citizens, parents, friends, believers to ideological systems. So I don't think it's wise to draw this division, because that also feeds to popular fears that we must have this regulation, because otherwise the scientists will go mad in the labs. But yeah, and then the like about the polls, and I just want to emphasize like how much the wordings and the available knowledge will affect how people think. Like just like imagine of having a poll about this, and then there would be some information box, and imagine of the embryo model of 28 or 50 days or whatever, like if it's in its real size in the information box, or an enlarged size where you can like really have a better view of the picture. But just I think just that would affect really seriously what people would believe about the embryo model. So it's really a matter of framing question and giving information, and that's a very, very delicate issue. Also concerning the fact that the one who kind of, if I would want to advance belief systems that would ban embryo models, and I would be making the poll, I would probably choose the very large picture because it would frighten people. But if I would advance embryo model research, I would probably select a very like a very murky and small picture, and like giving the information stating that calm down, nothing big is going on, it's all right. So it's a kind of self-fulfilling prophecy already when you make the poll. It's very hard to be neutral of like making research questions or like questions to the public. What do you think about this grave and big advancement that has all this stuff? Or what do you think about this mundane thing that we'll do in the lab? So it affects the answers.
Mm-hmm. Right. Well, we are almost out of time. So, yeah, I'm afraid we have to cut off the discussion right now because we have to end at 1 o'clock. So I'd like to just thank you, the panelists, very much for your excellent contributions, and invite Tuija Takala, the chair of, I was going to say president of, yeah, president is fine, of the NCBio Committee, to come here and say the final words.
Yeah, actually, just stay here, all of you. Yeah, just stay there. It's 2 minutes or less. So, anyways, I just would like to, on behalf of the Nordic Committee of Bioethics, I'd just like to say a few words to close this session. Basically, I'm just thanking everyone involved. Echoing what already Guðrún and Sigur said, I think we've had a really, really illuminating half a day. And I do like the two sides, so to speak, the scientist and the ethicist addressing a common theme. I thought that the balance of the talks was very good, and I very much enjoyed, especially the symposium as a whole. So in that sort of spirit, I would like you to join me in thanking the speakers once more, Jacob, Hanna, Fredrik Lanner, Garðar Árnason, and Johanna Ahola-Launonen. Thank you. And I'd also like to thank Sigur Kristinsson for his chairing of the panel discussion over here. Thank you. Then more generally, again, thanking the organizers and funders and all that, starting with Guðrún and the working group. Thank you. The whole graduate program, the BioMedical Center at the University of Iceland. Thank you. And Bettina up there and Nordforsk for providing much of the funding. Thank you. And organization. More generally, the Nordic Committee of Bioethics would like to thank Nordforsk for the past decade. And then finally, the Nordic Committee of Bioethics would like to thank all of you participants over here and online. I hope you enjoyed the day as much as we did. Thank you. And at exactly 1 o'clock.